Well, good afternoon, and a huge welcome to the Royal Society of Edinburgh. And can I thank the Scottish Government and Public Health Scotland and our partners for putting on what I think is a terrific event. So my name's Andrew Morris. I'm Professor of Medicine and Vice Principal at the University of Edinburgh, but I'm sort of exiled to London because I direct the National Institute for Health Data Science across the UK, and I'm also president of the Academy of Medical Sciences, which is one of the 4 national academies. But over the last few years, I've had the great privilege and pleasure to work with many of you in this room to really think carefully about how we responded to the recent pandemic, but also to look ahead in terms of how can we be better prepared next time. So we've got a great day ahead of us. But before then, because Marianne, she's written me a very careful note, I've got to do the fire drill. So there we go. These are the exit procedures. So the door is there, I think, or there. And apparently you meet outside the Dome on George Street. So Daniel Kleinberg, please resist the temptation to go in and buy a drink, okay? So that's the fire drill over. But just a few thoughts to kick off what I think is going to be a very important day, because it's 1,876 days since the first case of COVID-19 in the UK. And as you know, we sort of marked in a very somber way, appropriate way, the 5-year anniversary. I also note that Stephen Riker was on the BBC again today, so they're still paying his retainer. I think Stephen's here, is he? So we're reflecting on that, but we're also looking forward, and that's the theme of today. And if we look back, I would argue that in some ways the memories are dimming of the personal impact, the public health impact, the economic disruption, the educational disruption, the social challenges, and the global health inequity. But it's important that we don't ignore pandemics, because I think if we do, we forget pandemics, it will be at our peril. So that's a sort of theme of today. And I think today is about 3 things. Firstly, we're going to celebrate what we think, Cabinet Secretary, is a very good report. So here, says CMO. And can we thank, I think we should thank the people who did a lot of the work, which was Daniel's team, it was Marianne, it was Caroline who did a lot of the work. So we're here to celebrate the report. Secondly, we're here to celebrate great science. And because during the pandemic, I would argue, we got a lot of things, we struggled with a lot of things, but we got a lot of things right. And it's just to celebrate the science, because in my roles, I have the privilege of looking across the UK, and there's great talent in Scotland. It's just fantastic. And there were stellar global contributions from Scotland in how we understood the virus, how we understood the effectiveness of vaccines, surveillance, use of data, and we're going to celebrate that a little bit today. And finally, we're going to look forward and how, because it's how we're responsible to each other. How do we work as a community so that we're better prepared next time for the citizens of Scotland and actually make a contribution to any global response? Because it's not if, I think it is when the WHO next declares a public health emergency of international concern, I'm really keen that Scotland's ready to not only benefit our citizens, but also demonstrate that we can contribute to the global response, because it will be a global response next time. And finally, because context is important, so one of the things I do, I have the privilege of sitting on the Prime Minister's Council for Science and Technology. So that's a sort of premium scientific advisory group of the UK. And we met yesterday. And many of the themes yesterday are relevant today. One, the focus of data and AI. And how do we, in a trustworthy way, exploit that? Secondly, how can the UK lead on global science diplomacy? That's a key challenge for us as we see a threat to science globally. And thirdly, it's the regional economic growth agenda. So how can Scotland use its scientific assets to actually power the economy by working in partnership with government, the NHS, because we can't just allow the Oxford-Cambridge arc to eclipse the rest of the UK. So I think that this meeting is very timely, and I would thank you for coming. We're going to have a very open discussion about anything. Every comment is welcome, because we want to hear from everybody in the room. But I'll finish with a couple of quotes. So it was Mike Ryan. Do you remember Mike Ryan from the WHO? He said, "Be fast, have no regrets. You must be the first mover. The virus will always get you if you don't move quickly." So that's a theme of what we're going to do. But probably finish with a quote from Tony Fauci, actually, who said, "We will get through this, but we will have to help each other." And that's a theme of today, because today is around government, working with public services, PHS, working with the clinical community, the scientific community, the industrial science base, but most importantly, the publics, to be better prepared for next time. So those are just a few thoughts, and don't forget the fire alarm. But it's a great pleasure to welcome, we've got two fantastic speakers. We've got the Cabinet Secretary, and we've also got Paul Johnson, who is Chief Executive of Public Health Scotland. And this brief is so good, I can't see who's speaking first. Maria, is it? It's a Cabinet. I apologize, Cabinet. Could you join me in welcoming the Cabinet Secretary and to thank him and his government for everything they did and have supported our work. So welcome, Mr. Gray.
Can I say, Andrew, thank you very much. It's a great pleasure to be here. Can I say that's a better introduction than what I thought I was going to get? Because I got, and I was catching Andrew here saying we have a very good day ahead of us, but first, and I thought that was going to be my introduction, but first we're going to hear from the Cabinet Secretary before we get on to, the real business before us. But housekeeping was where you were going. So my appreciation, Andrew, for your introduction, the context that you've set the discussions in today, and my thanks to you for your chairing, and I'll come back to that in a second. It genuinely is a real privilege to be here today, and I'd like to thank the Royal Society of Edinburgh for organising this event and helping us to showcase some of the pioneering research that Andrew was speaking about, but also the initiatives that took place across Scotland during the pandemic, and which we continue to support us in building our preparedness for future pandemics. Almost 5 years ago, Scotland, along with the rest of the UK, was on the brink of entering the first lockdown in response to the threat that the COVID-19 virus posed. And I would like to take this opportunity to express my condolences to all those who lost loved ones and whose lives have been or continue to be impacted by the pandemic. I must also say at this stage that many people in this room contributed a huge amount to supporting ministers, government, and to provide public reassurance and messaging during the pandemic. Your advice on helping guide us through the pandemic was absolutely invaluable. And I say today, on behalf of governments and ministers past, but also present, my thanks to everyone that helped to contribute during the pandemic. We could not have got through what we got through without your incredible service to Scotland, so thank you all very much. Scotland's road out of the pandemic has highlighted the exceptional value to society of effective collaboration and innovation. It's therefore only fitting that these are reflected as key priorities in the final report of the Standing Committee on Pandemic Preparedness, which was published at the end of last year, and which today's event serves to mark the launch of. The report makes 5 overarching recommendations for how Scotland can improve its preparedness and ability to respond to future pandemics. Firstly, it calls for the creation of a pandemic partnership, which will connect the diverse expertise and disciplines needed to tackle pandemics. This partnership will become a cornerstone of the way that we connect expertise from Scotland's fantastic research institutions, our Public Health Agency and the Scottish Government, in order to protect our population and respond to future pandemic threats. During the pandemic, we saw many great examples of collaboration improving our understanding of the virus and its impacts. One example being the work of Professor Emma Thompson, who will be moderating our first session today, and whose research gave us a vital understanding of the timing and way that SARS-CoV-2 had arrived into Scotland. Second, the report calls for increased opportunities to promote that connection and collaboration, including through events like today, which build and maintain connections that are vital for us to lean on during emergencies. Third, it highlights the fantastic potential of data, and something that Andrew referenced strongly in his introduction. I know is something incredibly important to him, and something that we will collaborate on more in the coming period. And it also calls for action to ensure that we are well positioned to leverage our data assets in future pandemic responses. Scotland's use of data during the COVID-19 pandemic has been described by many as being world leading, and indeed it has been widely acknowledged that studies such as the EVE2 programme played a pivotal role in understanding of the pandemic and of the impact of vaccines once they were introduced. Andrew also referenced in his introduction the Oxford-Cambridge arc, the golden triangle, if you like, and how Scotland has an opportunity, I think Andrew believes this, to compete. I heard it referenced that Scotland has the opportunity to create a platinum triangle with the collaboration of our institutions here in Scotland. That came from Cancer Research UK. So we have a well understood world-leading research capability here in Scotland. Let's use it, let's collaborate and make sure that we can compete, and make sure that that economic benefit can be leveraged, but also the knowledge base can be leveraged here in Scotland. It's something that, as Health Secretary, former Economy Secretary, I'm very interested in taking forward. Fourth, it stresses the importance of innovation and continual horizon scanning. That innovation can take many forms, from cutting-edge biomedical research to exploring new ways that government and independent advisors can come together to inform our response to emergencies. The Scottish Government's Chief Scientist for Health, Dame Anna Domenechek, is a passionate champion of innovation in health and care, and the crucial role this will have for future pandemic preparedness, and is an absolute anchor for the government in terms of that opportunity that's there from the potential platinum triangle, the real triple helix approach to ensuring we are mastering and pulling together the various threads of strength that we have here in Scotland. Finally, the report underlines the need to ensure that inequalities are a central consideration of the way we plan for and respond to and draw on the behavioural sciences to support this, to support our response to future pandemics. We know that pandemics have a disproportionate impact on those already facing the most inequalities in society, and so it is all of our jobs and responsibilities to make sure that we try to minimise this as much as possible in future pandemics. I'm pleased that work is already underway to deliver on these recommendations, including, as we'll shortly hear from Paul Johnson, the work of Public Health Scotland that has been leading to establish the partnership. On a personal note, I know the work of this partnership has been a tremendous resource to support the Scottish Government's Pandemic Preparedness programme of work, including the Ministerial Oversight Group on Future Pandemic Preparedness, which I co-chair. I'm very grateful for that. So I'd like to end now by thanking the members of the Standing Committee on Pandemic Preparedness for their significant contribution. I would also like to thank Professor Andrew Morris for his exceptional leadership as chair of both the Scottish Government's COVID-19 Advisory Group and the Standing Committee on Pandemic Preparedness. The work of the Standing Committee and the creation of the partnership are a key part of our broader ambition to strengthen Scotland's resilience and Preparedness, working alongside our UK and international partners to ensure we are ready to respond effectively to future pandemics. And I very much look forward to working with colleagues in this room and further afield and making sure that we respond in the best possible way for the inevitability that is a future pandemic arriving on our shores. And I thank you all for all the work that's gone in for this celebration today of a publication of a report that I think takes us forward. And I look forward to hearing more about the collaborating opportunities that there are in the sessions ahead. Thank you very much.
Well, I'm glad we sorted out the running order there. That was definitely appropriate that you went first, Cabinet Secretary. Thank you very much for that. I would just get to the right slide there. I would just like to say how pleased I am, how honoured I am to be able to take just a few minutes at the start of our event on behalf of Public Health Scotland to add my own thanks for all the work that's been done in terms of the Standing Committee on Pandemic Preparedness report. The report is hugely welcome. And let me just say that from a Public Health Scotland perspective, we are now working to operationalise the recommendation in the report around the establishment of the National Scottish Pandemic Science Partnership. Indeed, very practically, a detailed paper is going to the Public Health Scotland Board next Wednesday, setting out the proposed arrangements for how it will operate and what role we will play. And we'll be very keen to share further details of that in the coming weeks. And I do emphasise we're talking coming weeks. I take Andrew's point about speed. We're not going to hang around and deliberate on this for months to come. The work has been done in the report, and I see it as our duty now to crack on and make sure that we can play our role in putting the arrangements that are recommended into practice. But I want to emphasise two key things, and the first is that as we play our role in Public Health Scotland here, I'm very, very keen that we do so listening really carefully to the expertise that is in this room and more broadly. So a number of us are here from PHS today. We absolutely recognise that we have a contribution to make in terms of the expertise within our workforce, but we must listen and collaborate with partners. That's what this Pandemic Science Partnership will be all about. So those of us here from PHS are here to connect with you and to listen really carefully to your perspectives, both in this crucial phase as we get the partnership established and indeed as we take it forward. But secondly, I think it's vital that we also reflect on the successes that we've seen in recent years and ensure that as we take the work forward, we build on those successes. So let me just take a moment or two to reflect on some of the work that has already been done over this last 5 years and ensure that we learn from that and take this forward into our future pandemic partnership. And it seems to me that what's absolutely clear, if we look at examples of real success through the pandemic, and Eve too has already been mentioned, we see deep collaboration at the heart of the approaches that have been taken. Public Health Scotland's predecessor organisation, Health Protection Scotland, already had good links with many universities prior to PHS's inception. But what we saw was the pandemic required us to build those partnerships much more fully and actively and work together, pooling together the specialist expertise that we have within our organisation as the National Public Health Organisation, but also recognising the many gaps that we have and our requirement, therefore, to work with all of you. And Eve too, I think, was possibly one of the best examples of an impactful partnership that really provided world-class leadership in demonstrating vaccine effectiveness. It truly was groundbreaking, offering the first population-level vaccine effectiveness estimates to the world from Scotland. Eve 2's submission was titled Real-World Validation of COVID-19 Vaccination Effectiveness through Rapid Use of Linked national data. And there we come on to a real asset that we have in Scotland. That is the richness of data that we hold, and of course, the requirement that we make very good use of that data going forward. And I can be clear that within Public Health Scotland, we recognise that the data we hold is a national asset. It's not for any one organisation. Indeed, it must be utilised to protect and improve Scotland's health. And that's what we will endeavour to do. The groundbreaking findings from EVE 2 ensured the continuing assessment of the effectiveness of the vaccine that followed for each of the subsequent waves of infection from different variations and from the revaccination of the population against these strains. And it's good just to reflect on some of the way in which UK leaders recognised the significance of the partnership work that many of you in this room undertaken, continuing here for a moment with EVE 2. There is Sir Chris Whitty thanking the analysts and investigators of EVE 2 and commenting that their analysis of Scottish data helped inform policy across the 4 nations of the UK and globally, as well as in Scotland. And in particular, it helped inform vaccine policy and public understanding of the likely impact of vaccines. And Sir Patrick Vallance comments on the brilliant insights that have guided not just the UK, but the world. I think it's appropriate we pause and, in that spirit of recognising success, simply acknowledge that we saw innovative partnership work flourish through the pandemic, and how vital it is that as we move into the implementation of the recommendations, we ensure that that doesn't just continue, but grows and flourishes all the more. If we can continue in our effort to recognise some of the successes, this, I have to say, of all the slides that my colleagues have worked on, and I want to particularly give a shout out to Professor Jim McMenamin in the room, who is the one who has, we particularly enjoyed chatting through this slide, and I think it might be Jim's annotations here in red, that's pulling out a particularly important fact. We all recognise, and will often, as we engage with the UK and Scottish inquiries, we recognise that not everything was done as well as we would want it to be. We recognise there is huge learning. But today we want to reflect on the effect of the marrying together of scientific leadership, government leadership, and open collaboration with the public and with professionals being so pivotal to the successful engagement with and investment by the public. The trust by the public was manifest in the rapid, high initial and subsequent revaccination uptake rates with COVID-19 vaccine by the public. And what this slide gets us into is the World Health Organization's estimates of the impact that these actions have. And what you see here is effectively a league table showing vaccine uptake and numbers of lives likely to have been saved through the work that many of you in this room did tirelessly through the pandemic. From a public health perspective, I find I'm often citing pretty miserable numbers. It's numbers about healthy life expectancy, and it's numbers about the gaps in terms of health inequalities, and they are really important. But there's a number here that I'd like us to keep in mind today, and it's the number 25,386, which is the World Health Organization's estimates of lives saved through the outstanding work that has been done by so many in this room and by your colleagues in ensuring that we reduced harm to the Scottish population, that we prevented deaths, and at the same time we tackled health inequalities. So there is plenty to celebrate today, and we'll be keen to continue to learn from the innovative work that has been done. The Cabinet Secretary has already run through the 5 recommendations from the Standing Committee on Pandemic Preparedness, so I will not dwell on this other than once again to welcome in particular the recommended establishment of a Scottish Pandemic Sciences Partnership and to ask for your ongoing engagement and contribution and feedback and challenge as we seek to take forward hosting arrangements for this work, but by no means seeking to see ourselves as controlling the work. Host. I see that as a modest contribution to something that requires a system-wide leadership endeavour. But let's be clear that there is already great work underway to learn from the pandemic. There is much learning yet to come from the UK and Scottish inquiries. But already within Public Health Scotland, we're working with the World Health Organization and the UK Health Security Organisation to draw on this important framework, crafting the mosaic for resilient surveillance for respiratory viruses of epidemic and pandemic potential. This is but one example of work that's underway today, being taken forward by a number of colleagues in this room. So again, I think we can take confidence from successes seen over the past 5 years. We can draw on work that's underway just now from within the organisations represented here, and we can commit to intensify our collaborative endeavours in the coming days as we take forward the Scottish Pandemic Sciences Partnership. Thank you very much.
Cabinet Secretary Paul, thanks for getting us off to such a great start. I should also have said, I think we've got over 100 people online, and folk online are very welcome to ask questions in the chat, and I think the terrific staff from RSE and Marianne will feed them into the discussion. So we do want this to be as interactive and virtual as possible. Well, I think that was a great introduction, and my response is let's do it. I think for the benefit of Scotland. I've been asked to reflect on what I've heard, so let's do it, is my sense. And let's start today. And I think what we've purposefully, because it's, I'm an old guy, we've purposely shaped today to hear from some of the, the really bright, energetic, early career researchers who are within the Scottish research community. So we're going to have a great time hearing from them and their research endeavours. But to take us through the first set of lightning talks, it's a great privilege to welcome my friend and colleague Emma Thompson from the University of Glasgow. She's acting director of the MRC University of Glasgow Centre for Virus Research. We've already heard about your own world-leading research, but it would be terrific, Emma, if you could take us through a few lightning talks just to give folk a sense of the breadth and depth of talent that we have in our midst. So welcome, Emma.
Thank you very much, and good afternoon, everyone. It's a real pleasure to be here today at this important event on pandemic preparedness. And I guess I might just say a couple of quick things before welcoming the young investigators, young scientists who are going to be really critical for us in the coming years. The last few years have obviously shown us in fairly stark terms how critical scientific innovation is, how critical scientific infrastructure and innovation are to global health security. And our route out of COVID-19 depended on the development of vaccines, the application of next-generation sequencing to track virus evolution, and the extraordinary and collaborative efforts of the scientific community to translate research into action at scale and pace. But our job is not done at all, there are perhaps as many as 300,000 generalist viruses that we have not even identified yet, which may have the capacity to cause pandemics of the future. And for that reason, and known viruses that we are responding to now, including avian influenza, which has caused a panzootic around the world, mpox, which is affecting many countries, and we are at risk of importations. This underscores the importance of sustained investment in scientific infrastructure and in innovation. And a key part of this preparedness is building the next generation of new leaders, new scientific leaders, training young scientists and equipping them with the tools and expertise to respond to future threats, which are a definite. We're going to have to respond again, is critical. And we're fortunate today to have some of those younger scientists with us. And I'll start now, and I'm really delighted to introduce the first of these speakers, Dr. Wael Kamel from the CVR, who is the recipient of a Moderna Fellowship to investigate RNA science. And I'll let you start. Thanks. Well, by the way, just to warn you, I'm going to wave at you. You've got 10 minutes each. So I'm going to be like really waving. Try to keep to time. I'll give you a signal if you're going over. Thank you. Thank you.
Hello. So I'm very happy to be here, and thanks for the invitation to talk about what we have been doing in the last few years. But I would like to start with a little bit about the motivation for our work and what is the idea that we had in the beginning, which started actually 1 year before the pandemic. And this is about what is the RNA, what we think when we talk about RNA. So usually people think about it as this linear molecule consisting of 4 nucleotides that will be repeated in a specific sequence. In fact, if we looked at the nucleotide that make the RNA, chemically speaking, they are very similar. And then if we compare it to proteins, for example, we see that the protein, we have wide range of amino acid. We have 21 amino acids which are completely different to each other. We have polar, non-polar, acidic, alkaline, and so on. And then these amino acids can form together forming domains. So we know that we have more than 20,000 different domains, protein domains. That's why you have the kinases, phosphatases, you have receptors, you have the membrane proteins. So that the main idea that people have in their mind that the proteins will be the mediator for cellular function, while RNA are just as a proxy for gene expression. But this is not really true, because RNA is not a linear molecule. RNA can be paired with each other, forming this 2D network, what we call a secondary structure. And additional interaction can be mediated by this nucleotide, forming a very sophisticated tertiary structures, sometimes as sophisticated as the complexes that we see with the proteins. Additional feature of the RNA, that's a shapeshifter. What do I mean by that? So for any given sequence of RNA, it does not exist as a single structure. It's usually as a probability of a different structures. And this shift between different structures can sometimes be affected by the environment of the RNA. For example, if you have a different metabolite that can interact with the RNA structure, it will favor one structure over the other. Or if you have a protein, so that can favor one structure over another. So why I mention this? Because what I want to tell you is that the RNA structure and its flexibility give the RNA a way to perform a very sophisticated function. So you can have an RNA that can act as a docking site for molecules, and also you can even have an RNA that can act as an enzyme, catalyze reactions. And what's really important about this flexibility part, that the RNA can change its function, or it can even regulate how it acts, its mode of action. And viruses know this long time ago. That's why viruses have RNA as a genetic material. So most of the viruses that now we are encountering, especially the one that we having a difficulty with, are having RNA as a genetic material. Why? Because viruses knew that if they have the RNA as a genetic material, they can incorporate so many different functions within the same RNA molecule. You have one RNA molecule that will be translated, transcribed, replicated, and it will carry out a wide range of interactions. And this balance between this process is not random. You have very tight balance between all of these processes, and if you misregulate this shift between these different processes, you can have increased virulence, decreased virulence, or completely inhibition of the virus infection. So that's the question that we wanted to answer. Can we actually capture all of these events that the viral RNA is performing? And one way to answer this question is by identifying the protein that interact directly with the viral RNA. So that's what we did. Back in 2019, exactly 12 months before we heard anything about COVID, we established the mesh without going in details. I don't want to talk about details or technicality, but what I will try to tell you is that we can, inside the live cells, we can label the virus RNA specifically with a photoreactive nucleotide. And this allows us at a specific wavelength, we can make the protein binding to the viral RNA fuse to it. So it will cross-link the proteins to the RNA. And then we can isolate the RNA with its bound protein, and then we go for mass spec, so basically quantitative proteomics. And we applied these techniques on SARS-CoV-2 with coronavirus, and we also applied for alphavirus, that's a different virus family. So these two families are quite different, but they have some similarity. Both of them are having RNA as a genetic material, and also another advantage that both of them replicate in the cytoplasm. So here we stain so the blue is basically staining the nuclei, and you can see in the infection, it happened in the cytoplasm of the cells in both cases. So what is the outcome of this method? So this method, what you will get when you apply this method, you will have two populations of the proteins. One that will bind to the viral RNA, one that does not bind. And the idea that we wanted to ask, are we enriching for relevant interactors with the viral RNA? Are we, for example, are we enriching for potential targets? Okay, so how we can answer this question? So we wanted to define something called master regulators. So what is the master regulators? Master regulators, that's what we define as the proteins that involve in regulating multiple viruses. So how we can find these ones? So simply what we did a meta-analysis for more than 70 genome-wide screen related to host virus interactions. So all of these screens are already published, and we just collected all of these screens. So that covered more than 37 different viruses, and across all of the different type of viruses that you can think of. And also we have more than 13,000 different phenotypes. And then we ask the question, the protein that binds to the RNA will be enriched in master regulators. So master regulators, that would be the protein that appear in several of these screens. So basically what we say is that master regulator, that would be the protein that involved in regulating more than three different virus families. And what we found is that the protein that interact with the viral RNA are actually enriched in these master regulators, while the proteins that does not bind to the viral RNA, they are not enriched, suggesting that we actually can capture a true interactor for a viral RNA, and they are significant for the viral RNA life cycle. This allows us to, first of all, to annotate the RNA binding proteins involved in the SARS-CoV-2 infection. And we can repurpose some of existing drugs against these proteins. So we allow us to discover a new therapeutic targets. But then we wanted to see, can we actually, using this method, can we actually discover new concepts or new insights for how the virus is interacting with the cell? And then we looked deeper into our data, and what we found was something that we didn't expect. So like I mentioned before, we work with the viruses that replicate in the cytoplasm of infected cells. So if you have an infection, you have the nucleus over here, and here is the cytoplasm, and usually the virus forms something called virus replication center in the cytoplasm. And what we found, we saw that there is a subset of nuclear proteins here. They move from the nucleus to the cytoplasm and interact with the viral RNA. Some of these proteins are part of a complex called the human spliceosome. So you guys, I think you know that the human splicing is happening by removing the introns from the pmRNA, then it will fuse together the two exons and forming this mRNA. And this process is strictly nuclear. And what we found is that the part of this human spliceosome is migrating from the nucleus to the cytoplasm upon infection. So then I can show you this. That's an example here. So here I'm staining for the nuclei. This is the DAPI staining for the nuclei. This is the nucleus. And you see this is one of these proteins. This is part of the human spliceosome. And you see in uninfected cells, it's strictly nuclear, matching the DAPI staining. However, once you add the virus, you see it started to radiate out of the nucleus to the cytoplasm. And it has this point localization, foci-like localization. And then if you waited for longer time upon infection, you will see that the signal from the nucleus is disappearing, and then you have very clear cytoplasmic localization. So what are these points? So it turned out that these points are actually co-localized perfectly with where the virus would replicate. So that means that this protein radiating out of the nucleus and try to find where is the virus and just sit with it. So why? So this is part of this complex. We call it the U2SRMB. It's actually called this. And I want to highlight that this is not just a protein complex. This is a protein with a piece of RNA. So this is called the non-coding RNA, the U2sRNA. And why I want to mention this, because the U2sRNA is act as a guiding device. So it's like a missile, and you have this guiding device on the missile, and it tells the complex where to sit. And we saw that this also non-coding RNA is migrating from the nucleus to the cytoplasm, and it directs the whole complex into the viral RNA. And when we try to interfere with this complex, we can knock down the proteins, we can interfere with the RNA, or we can even use a small compound that interferes with this complex. We see that the virus replicates faster. So we can see that the virus can, for example, when we look at the viral RNA, we can see increase up to 100-fold. And when we look at the virus titer, we can see up to 50-fold increase if we interfere with this complex. That will tell us that the human cells already have complexes that are dedicated to be antiviral, but also it can repurpose some of its own complexes that doing something completely different to be an antivirus in case of infection. So why this is important? Okay? Because so far, based on our knowledge, there are only 2 RNA-guided silencing complexes. One in bacteria that can silence viruses, phages, and that's the CRISPR. And then we have the RNAi, which initially as described as an antiviral in insect and in vertebrates. And here what we are showing that we show that the humans, or in human cells, we can repurpose part of the spliceosome to act as an RNA-guided complex. These 2 complexes already has been widely appreciated by the scientific community and have so many different applications. And we think that this complex also can have the same potential. But before that, what we're trying to do, we try to understand the detailed mechanism of how it's antiviral and how we can make it even more potent. So instead of having a 50-fold increase, can we completely have a 1,000-fold and so on? And also we can have targeting different viruses. Instead of targeting only Symbis, can we target other SARS-CoV-2, for example? And with this, I would like to end. I hope I didn't take too long. And I just the main take-home message, what I want to tell you, that RNA is not just a proxy for gene expression, and also by understanding the different aspect of the viral RNA, we can get so much insight that beyond basic science. And of course, this work has been done with the extensive support from the CVR and our long-standing collaboration at the Rosalind Franklin Institute, where they are expert in proteomics. And of course, this work was already initiated in 2019. We were in Oxford, and then we moved to Glasgow. And I would like to thank the funding bodies, the Marie Curie, the ARC, and the MRC. And thank you very much.
Thank you very much. Well, that was a very interesting talk, and we'll probably have questions, but we're going to do questions at the end of the whole session. So now I'd like to welcome Dr. Clara Calvert from the University of Edinburgh, who's going to talk to us about COVID-19 in pregnancy in Scotland. Clara, thank you.
Sorry.
Oh, yeah.
Thanks so much for inviting me to be here. In this very brief presentation, I'm going to give an overview of the COVID-19 in pregnancy COPS study, and I'll touch on a few of the main results to come out of this study, as well as very briefly reflect on some of the lessons we learned from COPS that I think could be useful for thinking about pandemic preparedness. So just as a bit of background, it was very clear from early in the pandemic that there was a real need for high quality data on the impact of SARS-CoV-2 infection on the outcomes among pregnant women and their babies, as well as on COVID-19 vaccine safety in pregnancy once the vaccines became available. In the absence of these data, we initially saw unclear guidance around vaccination for pregnant women. We saw fairly persistent vaccine hesitancy among pregnant women, and there was uncertainty around certain aspects of clinical management for pregnant women who had infections. So, for example, whether a woman who had an infection early in pregnancy might need more sort of closer monitoring during the pregnancy. There was huge potential for the data in Scotland to help address some of these gaps. And so the COPS study was set up by Rachel Wood at Public Health Scotland and Sarah Stock at the University of Edinburgh to address these gaps. And I got involved as to provide some epidemiological and analytical support to this study. So COPS essentially involved the linkage of routine electronic health data to create a cohort that included all pregnancies in Scotland from 2015 onwards. And this cohort captured really important information, including the estimated date of conception, the date at the end of pregnancy, the pregnancy outcome, and clinical and social demographic information on the pregnant woman. We were then able to link onto this information on confirmed infections, either just before or during pregnancy, and any vaccinations that occurred around the pregnancy period. And we were also able to link on data on 21 different pregnancy, maternal, and baby outcomes. And I'll touch on a few of those over the next few slides. Once we created this COPS database, we were able, the team was able to do some really important descriptive work, so describing the numbers of infections among pregnant women over time, as well as looking at vaccine coverage among pregnant women. And we were also able to do some more advanced epidemiological analyses. For example, looking at whether there was any evidence of an association between either infection or vaccination and any of the adverse pregnancy, maternal, or baby outcomes that we had in the data set. So I just have 2 slides with a few results emerging from this study. I'll just get my pointer. So this first slide, the graph shows the association between infection just before or during pregnancy and a selection of our pregnancy, baby, and maternal outcomes. So we have our pregnancy outcomes up in the top here in the blue, the baby outcomes in the middle, and at the bottom we have the maternal outcomes. And these analyses took account of other important potential covariates, so for example, maternal age or the Scottish Index of Multiple Deprivation as a proxy for socioeconomic status. And so an adjusted odds ratio of 1 suggests that there is no evidence for an association between infection and pregnancy and the outcome. And we can see that we do have some of our outcomes hovering around 1, but actually we did find evidence that for other outcomes there did appear to be an increased risk of these with exposure to infection and pregnancy. And specifically, we saw an increased risk of preterm birth among babies that were exposed in pregnancy. And for women that were exposed in pregnancy, we saw an increased risk of critical care or death during or just after pregnancy, and venous thromboembolism. So we did conclude that there was some evidence that infection was associated with some adverse outcomes for pregnant women and their babies. So a very similar graph, but this time our exposure is COVID-19 vaccination just before or during pregnancy. And what we found was our odds ratios were all sitting around 1. It looked like the risk of these outcomes was fairly similar in our vaccinated and our unvaccinated, or exposed to vaccination or not vaccination during pregnancy. So from this, we concluded that there was no evidence that vaccination was associated with an increased risk of any of the outcomes that we could look at within the COPS study. I just want to finish with this final slide, which is just a few reflections on what I took away from the COPS study. Firstly, I think it did produce some of the highest quality evidence we had on infection and vaccination within pregnancy globally. And that's in part due to the strength of the data we have within Scotland that have really allowed us to do these population-based studies. A lot of the evidence preceding this had been single institutions, hospital studies, so we could really look at this at a whole population base. Secondly, while the COPS study was very much a time-limited piece of work, it did actually build an important research resource for further work. So there is some ongoing work currently using some of the COPS work to look at longer-term child outcomes among babies that were exposed to infection within pregnancy. And I think really importantly to highlight was the COPS work provided a foundation for the development of the Scottish Linked Pregnancy and Baby Dataset, which is a national resource held within Public Health Scotland. And that now captures data for almost all pregnancies within Scotland from 2000 onwards and is updated on a monthly basis. So if we ever had to do a piece of work like COPS again, that would hugely facilitate that work by having that already in place. So that's an extremely useful data resource that's now available. And I just want to finish by highlighting, and I think this is a theme that's already come up, but this was one of the most productive collaborations I've worked on. And I think part of that was bringing together different teams, specifically from the University of Edinburgh and Public Health Scotland. So it was bringing together this in-depth knowledge of the pregnancy data and public health from Public Health Scotland with some more clinical and epidemiological expertise from the University of Edinburgh, but we actually also drew on our wider networks across the UK as needed. And I think that made this the successful project that it ended up being. And I will stop there and hand over.
So, thank you very much for that excellent talk and for both speakers for keeping really well to time. I'd now like to welcome Dr. Mark Atkinson from Heriot-Watt University to talk to us. Thank you very much. Let's make sure we've got your slides ready.
Yep, that's good.
There you go.
Okay, thank you very much. The pressure is now on for me to keep to time, thanks to everybody else. So, thank you for the invitation. As we said, I'm Dr. Mark Atkinson. I'm Assistant Professor in Psychology at Heriot-Watt, and I'm going to be talking to you about the psychological response that we need the public perhaps to adopt to help us during a pandemic. So, I'll start with the observation that a crucial part of the pandemic response will be asking the public to adopt unfamiliar, novel behaviours to reduce the transmission of the disease. So, in the context of COVID, of course, we required people to do things like physically distance, wear masks, self-isolate when required to, and even with the advent of vaccines, it's still important to have people adopting novel behaviours because we're asking people to accept an unfamiliar and quite novel, newly developed vaccine. So, at the height of the pandemic, I was part of a team of psychological researchers, and we were interested in what causes any variations in how much people are prepared to adopt these behaviours. So really we're asking, why is it that some people manage to adopt these behaviours to quite a large extent, and why do some people not adopt these behaviours when required to? And so we ran a series of experiments with thousands of people trying to determine exactly what causes this variability in the public response. And we were doing this with a view to try and find out how we could promote these behaviours from the public. And I'd like to highlight two of those particular studies and give you some overall findings of those results, which I think will tie in quite nicely from a lot of the other points made by the speakers so far today. So in this first study, we were interested in what causes the variability in behaviours such as physical distancing. Now, this was at the height of COVID-19 when we were carrying out these results, so we couldn't do things like invite participants into the lab and actually observe what they were doing in quite controlled conditions. The alternative is we could ask people what to do and whether they actually physically distanced, and there's a couple of limitations to this as well. One limitation is if you ask people how much they follow guidelines, they tend to tell you what they think researchers want them to hear. And another limitation is it's actually very hard to kind of work out how much you've been physically distancing from other people as you go about your day. So our solution to this was to complement asking people what they did with a series of quite intuitive online experiments, which look like little computer games, when we were asking our participants to consider themselves to be this blue little avatar, this little character here in the blue t-shirt with you written on it. And we asked them in a series of different scenarios to place their avatar on the screen where they thought they would position themselves in a series of different situations, a series of scenarios. And by varying the scenarios, we were able to see the relative effects of whether that person was positioning themselves next to a friend as opposed to a stranger, whether they were outdoors or indoors, whether people were wearing masks or not, or whether they were outside in the park or in a queue for a coffee shop, for example. And we also looked at the differences between standing physical distance and physical distance by asking people to do things like select a seat as if they're in a cinema or a doctor's waiting room. Now, there's a number of different factors which explain the variation in where people were placed in these avatars, but one of the factors that I'd like to draw attention to is their perception of the risk of COVID-19. Now, there's a number of different ways we can think about the risk of COVID-19. We can think about the risk of COVID-19 to ourselves and our immediate family, or we could think about the risk of COVID-19 to other people in our community. And what we found in these studies is that typically people will place themselves further away from others. They'll adopt these behaviors to a greater extent with greater perceptions of risk to other people in their community. Variability in how people saw the risk to themselves didn't affect the amount that they adopted physical distancing behaviors. So the implication for this is that if we want to promote these behaviors, we can do so by fostering a collective sense of identity in communities. And so asking people to act in the interests of the more vulnerable in their communities, as opposed to considering the risk to their individual selves. And I'll come back to this point at the end. Now, in the second study, we were interested in the role that providing support for these behaviors could encourage people to adopt these behaviors to a greater extent. And the behavior we focused on for this was self-isolation, because it's one of the more challenging things to ask people to have to do. And so what we were asking was, how do different types of support affect the amount of self-isolation that people would be happy to do and were able to do? And to do this, once again, we recruited hundreds of participants, and we asked them to imagine that they actually had to self-isolate that day. We gave them a writing exercise to make them really reflect on the personal challenges that they would face if they had to go through the self-isolation period. And then we gave them 3 different hypothetical levels of support, 1 to each group of participants. And so we either told our participants to imagine that they had no support to help them self-isolate, or they had a one-off payment of £500 to help them self-isolate, or for our final group, we asked them to imagine that they had £500 worth of support plus some practical support to help them self-isolate. And that practical support included deliveries of food and medicine if required, help with caring responsibilities if necessary, and if they needed it, the option of alternative accommodation while they were required to self-isolate. And this is a package that we saw in other nations in some areas, which was allowed during the pandemic. And because this is all quite hypothetical, we also ran a follow-up second part to this study where we only spoke to people who'd been required to self-isolate with COVID-19, and we asked them about the actual support they'd received to see how that impacted on their actual self-isolation behavior. Now, the key results from this study is that if you provide support, it can increase self-isolation in intentions, and it can increase self-isolation behavior, but crucially, only if that support is suitably tailored to people's individual circumstances. For some people, if you provide them with a payment of £500, this may increase the amount that they self-isolate, perhaps depending on their particular employment or financial situation. But for other people, a payment of £500 isn't going to have any effect. And the reason is this, is if they have caring responsibilities in particular, they need that practical support to enable them to self-isolate. So we've seen that tailored support can increase self-isolation, and from the data that we collected in these studies, we could also see how this had this effect. And actually, it has this effect through 2 different pathways. The first pathway is a very practical pathway. By providing with people with the necessary support to self-isolate, it reduces the barriers that they face when trying to self-isolate, which increases their self-isolation intentions and behavior. And the second pathway is a psychological one. If you provide people with tailored support, this improves their relationships to the authorities who are providing that support, and therefore increases their willingness and intention to adhere to the guidelines set by those authorities. So the conclusion from this is that an understanding and supportive relationship between the authorities and the public can increase preventative behaviours. So just to bring that all together for my final slide. If we need people to respond in positive ways to reduce the transmission of diseases such as COVID-19, one way we can do this is by having this sense of collective identities, these ideas of common goals and common purposes. And crucially, we need to build these among different members of the public, as we saw in that first study, and also between the public and the authorities, as we saw in that second study. So we need to foster this environment of trust, solidarity, and the necessary support structures to allow people to behave in the ways that we need them to reduce the transmission of disease. So it's just left for me to just thank my collaborators on this project, Professor Stephen Reicher and Dr. Fergus Neville, who are here today, and Vegangelis and Tontis, and thank you very much for your attention.
Thank you very much for another excellent talk, very interesting. And again, hold your questions. We have another speaker, and then we will have, I'm sure, a lively discussion to come. The final speaker in this session is Dr. Alice McLean from the University of Stirling, who's going to talk to us about qualitative research and long COVID. Thank you very much.
Thank you very much for inviting me along to speak today. This is a great opportunity to talk about our research. And today I'm going to talk about what we can learn about pandemic preparedness from qualitative research on long COVID. And specifically, I'll raise questions around our preparations for the potential emergence of new post-viral syndromes in the future. In early 2020, there were many unknowns about the new virus that was spreading across the world. Although understandings were rapidly developing, it was initially thought that COVID-19 caused either symptoms which quickly resolved or potentially life-threatening illness requiring hospitalisation. What became apparent in the summer of 2020 was that significant numbers of people who had been infected, including those with mild infections, were experiencing numerous debilitating and bewildering symptoms, leaving them unable to continue with normal daily life for weeks or months. This came to be known as long COVID, a term which was coined by emerging communities of people who gathered online because they weren't getting better, they didn't know why, and neither did their GP. Long COVID is defined as symptoms persisting for 3 or more months after infection and can affect multiple organ systems. It involves a wide range of symptoms which fluctuate in severity and impair the ability to conduct basic activities of daily living. Symptoms often include extreme fatigue, breathlessness, cardiac symptoms, cognitive difficulties, joint pain, dizziness, and more. And there's no typical long COVID patient because of how differently it affects individuals. Prevalence estimates vary across studies, but the most recent ONS figures show that 168,000 Scottish people reported having long COVID in March 2024. Of these, 80% said their symptoms adversely affected their ability to undertake daily activities, and 27% said their daily activities had been limited a lot. So today I'm drawing on findings from 3 studies. The first is a CSO-funded study of adults' experiences of long COVID. For this, 63 adults from across the UK were interviewed in 2021 and 22. The second, funded by the NIHR, explored family experiences of long COVID. This included 73 interviews with children, young people and parents, again conducted in 2021 to 22. Both of these studies fed into the creation of Health Experience Insights, or HEXI, web resources, meaning that anyone with access to the internet can go and look at video and audio clips of people talking about their experiences of long COVID. The third is an ongoing study, also funded by the CSO, which is exploring the impact of long COVID on critical service occupations, particularly teaching, nursing, and the police. So our team has written about the experiences of long COVID vanguard patients who were the first to identify the condition and seek healthcare support. And long COVID is not the first example of a new virus leading to a post-viral syndrome, and yet so many of our participants spoke of doubting themselves when their symptoms persisted or when new symptoms arose. For many, this was an isolating experience. This participant said, I was trying to work out what on earth is going on, and as a doctor myself, that has been really, really difficult. I can't imagine what it would be like as a layperson. The neuro stuff, the phantom smells, the tinnitus, nerve pain, all such vague and strange things. I didn't even mention them to my GP because I thought she might think I was completely mad. Another said something similar. I had no idea what was happening to me. Then there started to be reports about people having post-COVID syndromes or having episodes of COVID that just seemed to go on, which was just amazing, like a light in the dark, because I thought I was going mad. So what learning can we take from these experiences to inform our preparedness for future pandemics? Are there ways that we can embed the potential emergence of new post-viral symptoms within the disease surveillance and data collection systems that will be utilised as future pandemics arise? And some argue that this would help to better identify, define, track and respond to new conditions as they emerge. Our research also highlights the lack of awareness, understanding and acknowledgement of post-viral syndromes like long COVID. A key theme across our studies is the disbelief and dismissal experienced by people with long COVID, including when seeking help from healthcare professionals. This has been especially isolating for children with long COVID and their parents, when the legitimacy of the child's illness and parents' credibility are called into question. So this is an example in this quote where a mother was speaking about how people often question, does long COVID even exist? Is it even a thing? Is it not just that your daughter would have been poorly anyway? So not really understanding that COVID can do this and that she hasn't really recovered, like maybe I'm overreacting. So 5 years on from the start of COVID, interviews we have conducted very recently still feature disbelief and a lack of validation as standout themes. In our urgency to declare triumph over COVID and to consign all the difficult experiences it entailed to the pages of history, it seems somehow inconvenient to us to acknowledge the fact that many people's lives continue to be completely derailed by the long-term effects of COVID. We need to address this for people living with long COVID and other post-viral conditions now by raising awareness and researching mechanisms and treatments. And in preparing for future pandemics, we also must ensure that our plans factor in awareness of and preparation for new post-viral conditions. The COVID pandemic reinforced just how essential our frontline services are. Long COVID prevalence is higher among key workers, which is not surprising given these services are predominantly delivered face-to-face with the public. In preparing for future pandemics, it's important to reflect on the main challenges experienced by essential workers at the start of COVID, at the height of it, and since then. In the case of future pandemics, how can we use the most up-to-date knowledge and innovations to help us identify who will be most exposed and how we can protect them? Our ongoing study explores the impacts of long COVID on education, nursing and police services in Scotland. Early findings suggest that COVID and long COVID have significantly affected these services. Many interviewees with long COVID have not been able to stay in the posts that they had before becoming unwell, and those who are still working often struggle to manage. They describe these services as under more pressure now than they've ever been. We often listen to very harrowing stories in these interviews, and this quote, which I'll read out in a minute, is from a primary school teacher in their mid-30s, and it conveys something of the devastation that many people have faced. It's also just one example of the skilled and dedicated people who are no longer able to work in our critical services. I was the typical type A personality. I never sat still, never had a day off work. I was a perfectionist. I'd get up early, I would go to work. I absolutely loved my job. I was the principal teacher of infants. They were very much pushing me towards a headship, but I loved being with the kids. My life's completely unrecognisable to that now. I'm not well enough to work, and I have this feeling of being like a ghost. It was like I was there at school one day, and now I'm just gone. I think the hardest thing that I struggle with is people thinking that I'm just at it, or that I'm not trying hard enough, when I've never tried harder at anything in my life than to get better. So as well as understanding the extent to which long COVID continues to impact our critical services, we need to consider what we might do differently in future pandemics. Did we do the right thing in stopping access to free COVID tests among frontline workers? Did we think through the economic and social impacts of not continuing to vaccinate all workers in these services? And are we adequately monitoring the ongoing impacts of the longer-term legacy of COVID? And what needs to be done to equip and support frontline services now, as well as to be prepared to support us in future pandemics? So just to finish off, I think it's vitally important that we reflect on the challenges we experienced during the COVID pandemic and use the learning from all the data at our disposal to help prepare for future pandemics. And in this presentation, I've pointed to the importance of considering the long-term impacts of new viruses from the outset and embedding the potential emergence of new post-viral conditions in our responses so as to better track and understand them as they emerge. And just before finishing, I want to thank all of our participants for giving their time and precious energy to our studies. I also want to thank the research teams to see the research I've highlighted today has involved a huge amount of teamwork, and to also thank people with lived experience who have taken part in PPI work, which has helped to shape our studies, and also to thank the CSO and NIHR for funding the research. Thank you.
Okay, well, thank you to all the speakers for 4 wonderful talks, very diverse talks. I'd like to invite the speakers now to come to the stage and be ready to answer some questions. Questions can be made both here in the room and online. They'll be picked up online if you put them in the comments section. And also, if you're going to ask a question, please, in the room, please raise your hand and wait for the microphone to come to you before you ask the question.
I'm really sorry, I'm
yeah.
actually having technical difficulties online, and for some reason the comment function's not working.
Oh.
So please just give a...
Okay, I'm very sorry. The comments function isn't working online. If we restore it, I'll let you know. Otherwise, it's going to be us in the room. If you have a friend in the room, perhaps you can text them and ask them to answer ask the question. We heard some very kind of diverse talks. Firstly, Dr. Kamel with the RNA technologies and changes in science there. And I wonder, perhaps because we've got a mixture of people in the room who are expert and non-expert, if you might think a little bit about expanding on how this might lead to new sort of therapeutic interventions, and that, you know, the importance of the spliceosome, and how we might be able to interrupt that therapeutically. I think that would be really interesting for people to hear. And then we have also Dr. Calvert, who was talking to us about the vaccine studies in pregnant women, and maybe thinking a bit about how we recruit pregnant women in the future, in future pandemics, and when do we intervene and when do we not, given concerns about medicines in pregnancy. And then behavioural change. I loved the point that people care more about other people than themselves, perhaps. I think that's these sorts of insights are critically important, and I guess how do we provide more tailored responses in pandemics? How do we do that amongst the chaos of a pandemic and competing resource demands? And I guess these are some of the things we think about. And then Dr. McLean, the qualitative research and the importance of long-term consequences of infections. And I think we're learning more and more about that. It may not just be a problem in COVID, and it might well be a problem with future diseases. We certainly know other viruses can persist, including RNA viruses, and cause symptoms for many years. So I think probably there's going to be a lot to discuss. I guess you'll be thinking about these things. I'm going to welcome now the audience to ask questions. If someone would like to raise their hand. Okay, we'll start with the with the front row here. Thank you. Gregor.
So first of all,
yeah, wait, hold on for the microphone.
So first of all, can I thank everyone for their presentations? They're incredibly interesting. I'm going to. There's so many questions I could ask, but I'm going to choose one that I think is for Mark here more than anyone else, and it's. I think your case is really compelling that you described in the research that you've done. Misinformation and disinformation was something that we encountered quite strongly over that pandemic response with COVID, and it seems to me that that's likely to be an ever greater feature in the future. So did you have any thoughts as to how governments may in the future respond into that space to create that sense of common identity, common purpose in the people, when misinformation and disinformation is likely to be so rife?
Try and work out how to work the mic. There we go. Yeah, thank you. It's a good question. There is another area of work which specifically looks at misinformation from other researchers. So I haven't got all of that information at the top of my head, exactly what they found, but there is work going on. I think in terms of communication, I think there's a big thing to do with like building trust in terms of both showing that you have like very much supporting the public and you're giving them the tools that they need and the support that they need to actually enact these behaviors and show that you're very much on their side and you have these common goals. And I think also it'll be a matter of like behavior from the authorities as well. So it's not only kind of like instructing people how to behave in certain ways, but it's also making sure that they behave in those ways themselves. So showing that not only are you telling people what to do, what we might consider injunctive norms about how people should behave, but it's also providing those descriptive norms as well. So it's also actually behaving in those particular ways. And I think if you do manage to establish that situation where you do have a sense of trust and solidarity and common goals from the authorities and the public, I think it will be easier to combat misinformation, perhaps particularly misinformation online. But as I said, there's a wider amount of work on this, which is ongoing as well, and there's other researchers involved. But I think the sort of core principles of like collective identity and common goals is quite crucial to this.
Thank you.
Thank you. Next question, Daniel.
Thank you. My name's Daniel Kleinberg and I think about infectious diseases and pandemics, and I work in the Scottish Government. And I suppose I'm reflecting on the experience of working with the COVID-19 advisory group. So for each of your areas, there were experts who were helping us there, but we created those relationships during a pandemic and I didn't have them going in. So I suppose when we get to the next one, how close do you feel to government, to public health, to the decision makers? Do you see it as your role if that was to happen again? Because in a way, what we did was nationalise all of the universities in Scotland and ask them all to work for us for nothing. We've said thank you today, but probably not enough. So if it were to happen again, how willing would you be?
Who would like to answer that question?
Not currently planned.
One of our speakers? I'm happy to go first, but I've already spoken, so I don't want to hog it. But I'll do it anyway very quickly. I think we managed to put together research teams very quickly from people who wanted to respond and had different levels of expertise. And I think our experience of being able to put these teams together quite quickly, we've already got those networks ready to a certain extent. And I think the universities that we've been working for do have that experience of putting things together quite quickly. We're also able to secure funding quite quickly. So my project was funded by the UKRI. And by having these ideas about how quickly we can get funding, how quickly we can put things together, I think we've got that experience where we could do it quicker next time. And also, with a pandemic going on, I did feel from the academic community, many people really wanted to kind of like help. They wanted to kind of be contributing and seeing how their research could be applied in ways which could help. I don't know if anyone wants to add.
Add on something, but I would just echo, I think, a lot of what you've said, actually. I think, certainly from my perspective, a lot of the work that I had been doing pre-COVID wasn't running in the same way, so there was also a little bit extra capacity for people who had the right skills to be brought into COVID. And I certainly we developed a huge amount of networks during that time that I think we'd be able to draw on much quicker should it happen again. But, yeah.
I'm going to add something to this. I think that we all have to try to maintain working together and not retreat into silos. It's really critical. And there are other very pressing problems that we just don't see quite so suddenly, but which still exist. And working in parallel doesn't make sense. We do need to be more coordinated. And I think there's a willingness and we have to somehow capture the moment still. Okay, and another question, please.
Would it be reasonable to introduce a general vaccination for tuberculosis in Scotland? Because there's so many people suffering from tuberculosis and bringing TB from abroad, and COVID and flu are so harsh on these people. Would it be difficult to introduce that vaccination? Is it risky? Is it a risky vaccine that can bring problems to people, or what do you think? Would it be reasonable?
Okay, I have this feeling that our speakers may not wish to comment on TB vaccination, but let me ask, would you, or would someone in the room, public health official, like to comment on TB vaccination? Because these things are really critically important, and, you know, why we vaccinate people. Thank you.
Thank you. Yeah, my name is Claire Cameron. I'm a consultant working at Public Health Scotland in immunisation. So you're absolutely right. There are a number of different vaccines that are available that we don't necessarily offer to everybody in the population. So how we determine which vaccines are offered to everybody and which vaccines are only offered to some people is through the UK Joint Committee on Vaccination and Immunisation. And quite some years ago, it was decided that actually for vaccinations against TB, it is most appropriate to have that as a selective programme focused on those people who will benefit from it most. Within the UK, and obviously in terms of people that we then welcome to Scotland from other places, there's a limit to the influence we can have on vaccine programmes there.
Mm-hmm. Maybe that one we can continue that discussion later. I can see it's something that's on your mind. Returning to the panel, I'm actually going to ask my question to Wael, because I think that there are policymakers in this room who may not appreciate the sort of revolution in RNA technology that is happening and how that can be utilised to think about the real sort of scientific advances that could be done here in Scotland. So, Wael, can you try translating for, you know, maybe half of the room will be very familiar with RNA viruses and so on, and half not, RNA technologies and so on. We've probably, many of us had a vaccine made from RNA. Can you tell us a little bit about how your findings can translate in the future?
So I think we can see what we saw during the pandemic, that developing the vaccine took time. And during this time, many people died, right? So if we get another pandemic, it will also take time to make the vaccines. And the problem, especially with virus infection, that we don't have a lot of options as antivirus therapeutics. So what the main value from our work that we can, instead of developing a therapy, we can actually activate some of the antiviral complexes that we already have in our body. So for example, when we saw that part of the spliceosome can be repurposed as a targeting complex for the viruses, how we can make it more potent? Would it be possible to develop, for example, chimeric USRNB, like this small RNA that we found? Can we make a chimeric RNA that we can introduce as an mRNA, not in a similar way like mRNA, but it's very small. And then we can target it multiple viruses, for example. So that's the way that we think about. We need to, I think what's very important, what we saw, we need to have multiple options, and we need to have multiple options that actually works. Because again, we don't have a pandemic, and then we wait for 1 year until we get some sort of vaccine that might work or might not. So I think that's the main
This
thing.
presumably could be done in quite a targeted way. So if you've got the, you know the sequences that you're dealing with, and that treatments could be manufactured at speed potentially, although obviously there's a huge process for
absolutely
for licensing and so on. Deb.
Thank you. They were wonderful talks. I'm going to ask you something a bit more broad than either of your, I guess, any of your specific research topics, but you know, in order to generate the kind of high quality evidence that you've presented to us this afternoon, we need sort of really tangible career pathways for early career researchers, and having the standing army, if you like, of academics and clinical academics coming through. And I'm keen to hear from any of you, I guess, as to how you think, what do you think the future is looking like, and what can be done to make it better for academic career pathways and clinical academic career pathways, because that's critical, isn't it, to our future resilience, having people not just do good research here but stay here. So.
Absolutely. The panel, tell us about your experiences and, give us an answer to that.
Not sure where to start with this one. Yeah, it's challenging. I mean, I guess for full disclosure, I work now part-time for Public Health Scotland and part-time at the University of Edinburgh, and I found that that's provided me with more stability, but working just as an academic was incredibly insecure. And I think if there's any way to provide a bit more security, particularly through early career, mid-career, and a bit more continuity to allow you to kind of focus on the work that you think is important, I think that would be incredibly supportive, but it does feel quite challenging at the moment. But if you can make it through, it's a brilliant career that allows you to really contribute and do important work that you love doing. So, yeah, I do.
I think I would just echo that as well. It's thinking about continuity, and I think the difficulty is I'm not clinical, but and I'm not early career either. When I was delighted to be described as young. So I would say I'm more mid-career. But yeah, it's often a case of going from contract to contract, and sometimes you know there's the difficulty then of building your own careers. You often have to take what you can get within that. And yeah, so I've been lucky that I've had a lot of really good mentorship throughout my career, and I feel I'm in a really supportive environment at the moment, which is helping me kind of to sort of forge my own path. But it is very just not having that sort of continuity of you always have to think about the end of your contract and where your next funding is coming from and things. So it's not easy.
Okay. Any further additions to this?
I was just going to echo that as well and say this precarity and like short-term contracts is quite an issue that academics face. But I also wanted to make the point is like when we're kind of thinking about how to retain these networks and keep the people who are able to work on these things, because we have these temporary contracts, we do lose a lot of people, and we have lost a lot of people who perhaps would have wanted to stay in academia, but because of 12-month contracts and maybe having to move around a lot, they haven't been able to do that. So it's not just an issue of perhaps retention, but considering about what we could have had if we had more stable academic jobs. So.
And you can't just magic up
people
who are very skilled. No. It takes time and investment.
Sure.
I think Andrew has a question.
Yeah, so I thought 4 terrific talks. And actually, Daniel, I see the nucleus of a future Scottish Government expert panel here. So I'm just reflecting, and so I'm going to ask Will a question, because it was exceptional in the pandemic. We got a vaccine within about 9 months, whereas I understand previously the median time to a vaccine was usually 12 to 15 years. So looking forward, do you think that's going to be the norm now? And secondly, we've got new tools. So AlphaFold 3, Demis Hassabis and John Jumper got the Nobel Prize for AlphaFold. We now have an understanding of 200 million proteins. How will that impact upon your work?
So, I mean, definitely these new tools that we currently being developed, that will help a lot in understanding the interaction between the host and the virus. But I just want to go back to the vaccine. So the vaccine is still not a universal solution, because there are so many different viruses that have no vaccines. Like it worked for SARS-CoV-2, but still we don't have vaccines for so many different viruses. So just relying on one option, I don't think that would be the best strategy to follow. And we need to deviate in our antiviral therapies. AlphaFold, that will work very well, but I think the main point what I was trying to point today that there is oversight of the importance of the RNA itself, because, and I think this is area that's not explored at all, like how we can look at the RNA structure and we can see some of the opportunities for therapies intervention in this area. And that's what actually I was awarded a Moderna Fellowship a few months ago. Hopefully we'll resolve it too. And this is basically the basis for that, how we can look at the viral RNA and look for important structures that we can target, or at least we can learn from it what is the function of these structures. Thank you.
Those are going to be critical. Yes, question just here on the side, and then I'll come over.
Tin Shi from University of Edinburgh. I have a question for Will as well. It's a quick one. I'm wondering, what's your thoughts about the combined vaccines?
So I just want to say that I'm not an immunologist. So I don't want to say, maybe Emma can say more about that because...
I'm the chair. Yes, I think combined vaccines are really important, and there'll be opportunities to do that. And also, I think also Weil kind of mentioned the fact that viruses are very diverse, and they escape, as we know, with each of the variants of COVID. The vaccines became a little bit less effective and had to be adapted, and we see that, of course, standardly with influenza. It may be possible in the future to put two or more variants into a vaccine, and that, in fact, we're already doing that, of course, but we may even be able to do that at a higher scale. And I think that is likely to be important, particularly with RNA viruses, which are very, very variable. So, but they need to be evaluated, and that evaluation does take a bit of time. You can compress the bureaucracy, and you can do the science at pace, but the testing for safety and efficacy always takes time. Next question. Chris.
Thanks. I had a question for Alice. So one of the key things in the pandemic is working out what decisions to make and what is the impact of a potential public health intervention. And we've mentioned already the way in which data has facilitated that to be done on vaccination in Scotland. But it seems like long COVID presents a particular challenge, because if you're looking at how an intervention affects, say, hospitalisation with COVID, hospitalisation is something it's very easy to know if somebody's in hospital or not and why. And also people tend to be in hospital quite quickly after they're infected. Now, mentioning long COVID, it seems that it's a very significant health impact. It's very complicated and it takes quite a long time to emerge. Are there ways in which that kind of impact can be understood quickly in the case of a new pandemic? Because you could be making decisions, if the real impact is not something you understand straight away, can that be accounted for, understood quickly? Are there ways to... Does new disease X have long disease X? Are there ways in which those kind of impacts can be brought into kind of epidemiology, those kind of ways of thinking?
So I think ideas around that are just starting to come out. There's some interesting papers come out recently about how to do that, and it's about embedding the kind of the possibility of something more longer term in your disease surveillance as, you know, at a very early stage. So having that, the possibility of something longer term developing, having that possibility in your mind when you're designing all your kind of ways of collecting data as a pandemic arises and as it continues. So yeah, I mean, like I say, some ideas about that coming out at the moment, just learning from long COVID and how that sort of developed, and it was fairly early. I mean, it was around May 2020 that people started to sort of talk about long COVID and not getting better and things. So it could, you know, it could have been, we could have started to think about it at that stage. But yeah, I think these ideas are at a fairly early stage at the moment.
Thank you.
Thank you. I think we have time probably for one more question. And I know this gentleman's been waiting patiently. Yes.
Yeah, great, cool. Yeah, thanks. I can't see behind me. Cool. So yeah, thanks very much everyone for some really great insights. This is a question really for Mark and Alice. It's about, Alice, you sort of mentioned, you hinted towards like a sort of societal disbelief of people with long COVID, or perhaps even institutional disbelief of people experiencing long COVID. How do we think we can challenge that sort of that medical institutional sort of disbelief and foster sort of feelings of solidarity between institutions and people who are experiencing those problems?
I think a lot of it's to do with awareness. So we need to really work on, I mean, I think it's understandable in many ways that we have had this emphasis on COVID and that we kind of triumphed over it and that it's now over and people want to move on, and that's understandable. But for many people, they haven't been able to, and their lives are completely destroyed. So it's the awareness of that. I think it's just inconvenient for people to think, oh, actually, this, you know, for some people, this is awful. And we need to raise awareness so that there can just be more acknowledgement and more just understanding, because that's what really people want, is just not to be faced with the disbelief again and again. So that's one way. And I think also in terms of, I think definitely things are getting better in terms of medical and healthcare professional interactions. We have seen that a bit across, because we've been doing these interviews for a number of years. So some people have had really supportive GPs, and one of the really supportive things that a GP can say is, I believe you, I just don't necessarily, you know, we don't necessarily know how to treat you at the moment, but I will listen to you, and if you ask me to look up something, I'll look it up for you. So that one of our participants described it as co-experting with their GP, because they could have that conversation and they felt really believed. So I think we need to get that also into education for healthcare professionals is the importance of that, and to have long COVID and other post-viral conditions more on sort of curriculum, so that that can be more embedded across.
Lovely. Okay, I think we need to wrap this up there. Thank you very much for all four speakers, for all the comments, and I'm sure there'll be room for more discussion in the coffee break. And before that, perhaps we can give them one more round of applause. I'm handing back to Professor Morris. Thank you.
Do you want the fire drill again? All we want to do, I think, what a great panel, wasn't it? And I think what we're seeing is a fusion of social science, behavioral science, computational science, and it's just wonderful. And the more permeable we are across disciplines, the better, and the better science. And Scotland's a great place to do this. And I think you've seen that from these four wonderful talks. So thank you very much. And coffee. And we're coming back in about 20 minutes, but in 3:45. So enjoy your coffee. And again, thank the Royal Society of Edinburgh, because it's going to be great coffee. Let's go. Are we? Okay. Oh, shall we kick off? So, what a great first session, wasn't it? I thought it was fantastic. So, our very esteemed panel has got a lot to live up to. So, but I'm sure we're going to have a great hour's discussion, and I'm delighted to invite Anna. So, Professor Dame Anna Dominicek, who's the chief scientist, to come and chair. I've actually known, I was reflecting over coffee, I've known Anna since 1990. So many of you weren't born. So, and I remember in the early days.
I was still in the nursery.
So, but in those days, because I worked in the Glasgow Western Infirmary, and Anna was my registrar. So, and we had a very famous surgeon. I won't tell you his name, but in those days, appraisal was very different. You know, today appraisal's just acres of forms and stuff. So my first ever appraisal as a young doctor was this surgeon says, "Morris, come here." So I went walking up the ward. He says, "I've got good news for you. This is your appraisal." He says, "You've gone from being completely useless to useless." And that was it. And so Anna was my registrar. I've learned a lot from her over the years, and please welcome her, because we're going to have a great session on innovation. Anna.
Thank you. Right, thank you, Andrew. I think I was a freshly minted consultant, and you were a registrar then, and knowing Andrew's dedication to research, he was always having a patient in research unit. That was it. So that's good. That's my memory. But of course, as somebody famously said, recollections differ. Yes. Right. So we have a session, we have an hour to talk about the power of partnership, academia, industry, and public sectors in action. This is my favorite topic, as I said, I think, to Marianne some time ago, and that's why I am here. We have one colleague couldn't join us, so we have 3 great panelists. But before we go there, I would like to take you, both colleagues who participated hands-on and others, to 12th of March 2020 for just a few minutes. So on the 12th of March 2020, I received a phone call very early in the morning from a colleague I worked very closely with, Chief Executive of Greater Glasgow and Clyde Health Board, who said to me, "Anna, I need tomorrow 5,000 COVID tests, and I can do 200. Could you help?" And that was the beginning of Lighthouse Laboratory in Glasgow, and then a network of 10 Lighthouse laboratories across the UK that I had the privilege to look after and help develop. But none of this would have been possible without collaboration. Collaboration, as per our title. We had engineers and architects who came to change the laboratory in the middle of all this in Queen Elizabeth University Hospital. They removed walls, they put things in. We had colleagues from universities in the west of Scotland sending us molecular equipment so we could do tests. Lots of equipment from research laboratories that, of course, were closed. We had industry, and that was absolutely vital, as we just discussed with Christine and others over coffee. There was Thermo Fisher Scientific, a big international company with headquarters close to the airport, who came with their facilities, with everything they had to help us. There were 2 SMEs, BioAscent and BioClavis, that actually ran the laboratory. Without them, we wouldn't have done it. But most importantly for me, there were 800 volunteers from local universities, mostly University of Glasgow, but also other West of Scotland universities, mostly PhD students, postdocs, who came. We couldn't take them all. We didn't have space. 800. The data do exist. And I think we need to appreciate this, that this triple helix collaboration was absolutely key. And without it, nothing can happen in the pandemic. So can I ask the three panelists, Matthew Holden first, then Poonam Malik in the middle, and Deborah Williamson here. We have given them some prompt questions. Marianne did a lot of hard work. So we have given them a couple of general questions. But what I will do now is I will give you a short bio of each of our panelists. And then after this short bio, they will tell us their 3 minutes of important things. And then we'll proceed with questions. So are you ready? Yes, they are ready. That's great. So we delighted to have Matthew Holden with us. Matthew is a professor of pathogen genomics and head of the Infection and Global Health Division at the University of St Andrews. He's also honorary consultant with Public Health Scotland, and he's leading pathogen genomics efforts for Scotland. He helped to establish Scotland's COVID sequencing service during the pandemic and integrated genomics into the Scotland's pathogen genomics strategic plan after the pandemic. Matt, the floor is yours for introductory remarks.
Yeah, thank you very much. I think the sort of introduction you've given me sort of gives a good potted history of sort of my background and also what I've done over the last few years and perhaps why I'm sat on the stage here. Just hearing the comments you started with and acknowledging the sort of Lighthouse Labs, I think just shows the power of the collaborations and how the sort of communities, whether that be within public health, the NHS, industry, or in academia, stepped up. And I think my own journey through that was as somebody who was a research scientist who was very much interested in using genomics as a tool to study pathogens and had some role within PHS before the pandemic, just as an advisor. There was an opportunity to, when all hands on deck came about, to actually step up. And again, for me, as a scientist and a researcher, a tremendous opportunity to feel like I could contribute, but also to help in the development of genomics and unleash the power of it during the pandemic. I think clearly in the pandemic, we capitalised on COG UK, and I think we'll perhaps reflect on that a little bit more, an academic partnership across the UK that stepped up with key partners here at CVR and also the University of Edinburgh. And my role...
I haven't touched anything.
Okay.
Oh dear.
Shall I try again? Oh.
Maybe we have too many mics on. Somebody at the back could help us. I think it's stuck. I think it's okay. It's okay.
So my role was to sort of integrate that into the public health response. I'm working closely with colleagues across NHS sequencing labs and the teams within PHS and also more widely within government to actually make the genome data useful, ensure that it was getting to teams that could use it, but also interacting very closely with the research teams to enable them to get access to the data to bring new insights into it. So I think that was certainly for me one of the most, I think, probably defining moments of my career, and I feel privileged to have been a part of it.
Great. So we move to a second panelist, Dr. Poonam Malik. She is an entrepreneur, academic, and business strategy leader, so whole triple helix in one person, with extensive global experience in the research, innovation, health, and business sectors. She is chair and co-founder of Microplate DX Ltd, the company that got many, many prizes over the last few years and is really well known across Scotland, UK, and internationally. She's also a climate champion for Scottish Enterprise, She used to head the investments at the University of Strathclyde. She led entrepreneurial and commercialization efforts for startups and spin-outs. With expertise in life sciences, biotech, and health tech, she holds MBA in strategy and leadership from the University of Edinburgh and PhD in biomedical sciences from the University of Glasgow. Please give us your introductory remarks.
Thank you, Anna, and good afternoon, all. It's a pleasure to be here. I think, as you said, today's topic, so I am honored to be discussing the three pillars, the triple helix, which is very close to your heart, as you rightly said. My PhD is from Center for Virus Research, Glasgow, many moons ago, and continued the research. So when the pandemic hit, it did bring back the memories, and it also highlighted the importance of science plus its interaction with society and communication, which we will discuss probably during the phase. But also, what it also highlighted is that the RNA research topic was discussed. This technology existed in 2013. The company had it. They were struggling to find investors to invest in it. So that indicates the role of business plus the role of society and investment to say what do we prioritize. And so is it reactive or is it proactive? Until and unless it hits us, we are not going to invest in because we don't see the value of it. And when it hit us, suddenly we are trying to catch our tail. Sorry to be on that front. So in that case, there is that future foresightedness, and which is why we have experts who are doing great science. And I have the pleasure of understanding science and that virologist background. But as Anna said, when on that day, when the 12th of March, the phone call comes, then also the other key aspect later on, we have seen all on the TV, those pictures of when in Italy hospitals, patients were in the corridors. And we were thinking, and my husband's NHS pediatrician consultant, and they always are prepared because it's cystic fibrosis. So the children initially were okay because they were taking all the precautions. It was the adult population that was the problem. But in that case, we all needed ventilators, and we didn't have. And that's where the value comes of another segment, which is industry, engineering, and they came together, and there has been wonderful example of collaboration. So if I were to take that learning from that, you understand science, but the true collaboration of government coming together, excellent science that was there, scientists, community, and volunteers collaborating, but the business fitting it in. And at the heart of it, eventually what boiled down is, the government de-risked it by putting some money on the front and the table and needs to do it, but it's the willingness of people that came. So the question for today, and every anniversary today is the 5th one we think about is, do we wait for another pandemic to come or do we do certain work? And I think during the discussion, we look forward to talking more about that.
Great, thank you very, very much. And our third panelist is Professor Deborah Williamson, who is a Dean of Medicine at the University of St. Andrews and a clinician scientist with extensive experience in both clinical research and public health research and activity. Deborah was in Australia where she led national reference laboratories, so has extensive experience in what we're discussing today. In addition to her current academic role, she also holds a position in the Public Health Scotland when she leads the Public Health Microbiology Division. She published more than 300 papers and received many awards, example being the L'Oreal, I can't say it, but UNESCO Women in Science Fellowship. She returned to Scotland in 2024 via a stint in UK HSA in London, where she, I think, inherited my team, and once I left UK HSA, and we shared some wonderful colleagues there. She joined University in St. Andrews and Public Health Scotland, and we're delighted to have her on the panel. Deborah.
Thank you, Dame Anna, and thanks for the opportunity to be on the panel today. As Anna said, I'm a clinician scientist who specialises in microbiology, specifically public health microbiology. And I trained in Glasgow, and just to close the circle of life, so to speak, I was Dame Anna's medical student at the Western Infirmary. Makes me really in the mid-1990s, and I did actually, Dame Anna may not recall, as recollections differ, but gave me my very first laboratory-based project, and I was delighted to spend the summer in a darkened room off Byres Road, scanning rat kidneys under a scanning electron microscope. So thank you for that, Dame Anna. As she said, I worked in public health agencies and academia across various countries, most recently Australia and the UK. And in that sense, I guess I'm a bit of a muggle, in as much as I've had one foot firmly in public health and the other in academia for many, many years. And I've seen I guess the tensions of the two, where people are working towards the same outcomes, but on different journeys there. But I've also seen the strength of collaboration between academia and public health. And when you hit that sweet spot, as I think people have in Scotland, and I think Emma's a great exemplar of that, it really works incredibly well. And I'm very hopeful about this partnership really, hitting that sweet spot of collaboration. I guess one thing I do want to say, and for me as a microbiologist, the two most chilling aspects, I think, of the pandemic were, one, the speed at which the virus took off. I mean, it was really frighteningly quick. But second, and I think perhaps somewhat unforgivably, the speed at which we seem to have forgotten how quickly it took off. And we talk about pandemics being a once in a generation, but what we also forget is that the way that we live now makes another pandemic much more likely, whether it's global connectivity, urbanization, climate change, the way we produce our food. They all increase the risk of pathogens emerging and re-emerging quickly, and before really we're able to catch up. But we've rapidly disestablished many of the structures we put in place, and we do need to think about that. And that includes things like research funding mechanisms as well. And as Dame Anna said, when I was at UKHSA, one of the sobering things that I was involved in doing was being part of the team that oversaw some of the mothballing of those laboratories. And it was not an entirely pleasant experience, let's put it that way. So again, I'd like us to think about having those insurance policies for next time. Thanks.
Thank you. Thank you. So you heard from all three panelists. Now you are thinking about questions, but we will give each one question in turn just to warm them up for you. So the question number one goes to Matt. And Matt, your question is, could you explain the role of whole genome sequencing in detecting, tracking, and mitigating the spread of infectious diseases, and particularly epidemic, pandemic of the sort we're talking about?
Yes, certainly. By way of an introduction, I think it's worth just reflecting on one of the legacies and one of the benefits of the pandemic is the investment in new technologies and new capabilities we have in Scotland, and genome sequencing is one of those. Before the pandemic, we had very limited capacity, and we lent very heavily on colleagues in academia to actually provide that through COG and indeed the centres we collaborated with in Glasgow and Edinburgh. So we've been very fortunate to get the investment in genome sequencing in Scotland to build up our capacity, and not just the sequencing itself. And arguably, the sequencing is the easy bit. It's the bits that go around it. So the bioinformatics, the training, the actual dissemination and thinking about the reporting. So it's there, and we're going to be expanding it over the next 5 years. We have a strategic plan, and that capacity is there for pandemic preparedness. So hopefully next time round, we'll be better prepared. That said, the benefits and why we're so excited about that capacity is genome sequencing is a fundamental technology that allows us to track and trace the spread pathogens. So put simply, each living organism, and indeed viruses, have their own genetic material inside them, and genome sequencing is effectively taking that molecule and decoding it. And typically, we decode it and put it into a digitized format that we can analyze using computers to extract the information from that and share that rapidly around the globe, so it can facilitate the sort of sharing of intelligence. But the information that genomes contains is very, very powerful. I think the talks we had this morning, particularly the talk about the RNA, illustrates by looking at the genetic material, we can understand how pathogens function, and also their weak spots that we can target, whether that's through a vaccine or indeed new therapies. But the power of the genome sequence is it's the genomic inventory. It contains the instructions for life that tells us about how the pathogen may work or survive and thrive. And again, it's the Achilles heels that we can target. But also it's a historical record that it contains information that's from its sort of parents. And so we can actually use that information to track and DNA fingerprint pathogens to work out their sort of origins very readily, whether that be on a ward in a potential outbreak and identify the spread in a closed setting, or actually track their spread around the globe. And in the case of SARS-CoV-2, we were faced against, as Deborah mentioned, a pathogen that spread very, very rapidly. In the first month of the pandemic in Scotland, and I think Emma will correct me if I'm wrong, was it 284 separate introductions into Scotland? And we were able to tell that from genome sequencing, and it was very powerful information that allows us to track pathogens almost in real time. So it's giving us really powerful information to inform our policies and also our practices.
Right, thank you very, very much. So Poonam, one for you. What challenges do researchers and entrepreneurs and industry colleagues face in turning innovation, discovery into real-world solutions?
Thank you, Anna. I think that's innovation is the life and blood of the new technologies coming to market and new ways of doing. It could be a product, it could be a service offering, but that needs to be innovate. And as you rightly said, in terms of our scientific excellence, research publications, grant securing student papers, we are world leading in the world. Sort of Scotland, to say the same knowledge that we punch above the weight is a cliched sentence, but the reality is in translation of that knowledge, we are not that far ahead. There are countries who do. The question is, why are we not able to translate that knowledge that we have acquired into something that could... And UK is a global leader in science, but to make that and to turn into an economic opportunity where we are able to scale and grow these companies and then deliver the product which are game-changing, something needs to change. And that's where the real power and real challenges come. So in terms of that, the requirement is in terms of the focus of the universities, yes, blue sky research and focus research. But at the same time, if we are living, the NHS is facing challenges, then instead of us getting technology which have been proven elsewhere and which are developing, we probably are needing to work on those challenges. So the university researchers that are working towards that, where is the knowledge gap? Is the intellectual property being secured early? Are there pathways? Do our PhD students and postdocs and even academics have that entrepreneurial knowledge? Is it part of our students, even any course they join, let alone medicine or health or engineering or computing or what the science, whatever background is, and even humanities and creative? The question is that do they understand what is required to turn that into, and including that early part will start to think about the commercialization. And not every discovery need to turn into it, but there are some life-altering and changing ones which we need to be, because if we have the knowledge. As a result of that, then what is the paraphernalia? Some universities are now being advanced in there, but others find because a company creation requires an external factor which academics are not prepared for. So you need business mentors, you need people who have been there, done that, mentoring, business planning, where the funding is, engaging with lawyers and other accountants, which is not everybody's cup of tea. So to do that, that structure is required to be in the universities. And as we have seen currently, that there are challenges that are facing higher education sector. But at the same time, this is a need for happen, because otherwise we don't want to be losing our world leading position in that we don't take our discoveries and invention to the World Forum and lose out. We don't want to be an incubator economy. That's the other thing that's been called for UK, that we develop these early stage companies, then we either lose them elsewhere, and that's the main problem, which I haven't even mentioned, is the funding, which is early stage funding or venture funding and scale up funding. And there are different challenges. So multiple challenges, and happy to discuss with them. But for now, I will keep it at that, and then probably, hopefully, next one.
That's great. And the third question goes to Deborah. And you've already told us that you have extensive experience of several public health systems, including international, UK, Scotland. Clearly, collaboration is key, and we knew it really well during the pandemic. Do we still have it? Do we collaborate enough? Is this all connected? Because when it happens next, it has to be connected.
Yeah, thanks. And, you're right. I think successful collaboration, it hinges on trust, it hinges on shared objectives, and the rapid exchange of data and knowledge. And trust cannot readily be built in a crisis. So strong, formalized relationships between government, academia, public health agencies, the NHS, industry, these should be established before a pandemic. These are peacetime activities. An informal goodwill is not enough. So structured agreements, data sharing frameworks, joint preparedness exercises. So these are all things that ensure operational readiness. And we talk a lot about preparedness exercises, but often they're done in silos. We don't think outside the box about bringing the public into preparedness exercises. Heaven forbid. So these are the sorts of things that we need to be thinking about as well. The other thing I think that's really important in collaboration is, and it's slightly nuanced, but it's about humility. And it's about respect for what others do. And we heard, I heard anyway, a lot of purisms and absolutisms in COVID. We can't possibly use LFDs. They're not as good as PCRs. Well, to quote Mike Ryan again, perfection's the enemy of speed. If you've got a test that's 80% sensitive in the middle of a public health emergency and it's cleared by regulatory agencies, why not use it? So, and I guess another absolutism I heard was, we can't possibly do that kind of research. It's not an RCT. Again, that's rubbish. Research is a continuum, and we heard some excellent talks, before the break about different kinds of research. But in order to do that kind of research, we need the right funding mechanisms, and we need them to be in place before a pandemic hits. So, yes, collaboration, but it's built on formalized agreements, and it's built on trust, humility, respect.
Great. Thank you very much. Now, they are now open for your questions, and I understand that also we can do something online. No, we can't do online. Technology, right. We see the question here, and could we get the microphone? Please introduce yourself once you have the microphone. Thank you.
Hi, Anna. Mark Woolhouse, University of Edinburgh, Professor of Infectious Disease Epidemiology. Thanks very much to the panel. You've all said how important it is to have the systems you're talking about in place and maintained in peacetime, which is absolutely right. But Deborah, you also mentioned the speed of events. And I just want to underline the challenge that presents and see what the three of you think about it, if you don't mind. What happened in Scotland and elsewhere in the UK in 2020 was very much determined by what we did and didn't do in January and February of that year. That set an awful lot of the path that we were then forced to follow. And it's wonderful that we now know there were 284 introductions of the virus. Thank you, Emma. But we didn't know it at the time. And we didn't know a lot at the time. And we didn't have the situational awareness that we needed to make the right decisions at that period. So we made a lot of wrong decisions in that period in the end, but out of ignorance, not of anything else. And I'm very pleased that some of the initiatives that people in this room have been involved in have come in for such high praise this afternoon. Eve, I was a member of that. It was led by Aziz Sheikh, of course, who takes a lot of the credit for setting it up. But Eve didn't get going until June of that year. COG UK, I think, Matthew, was about the same time before it actually started delivering a little bit. March, well done, but not in January and February, which is the point.
Of everything.
So how do we get to the position where, and it wasn't just those two, and there were many other initiatives, React, Comix, that provide absolutely vital data. They weren't operating in that very early stage when we needed them. So how do we do that?
Right. Deborah wants to start.
Yeah, thanks.
Great.
Thanks for that great opening question. Two things. One, and this is a reflection, because I spent the pandemic in Australia. I was out in Australia there, and obviously quite a different pandemic to the UK, in as much as for most of the pandemic, the public health policy was keep it out, not manage cases, but keep it out. Yes, and I think the single most important, and that stayed the case for all of 2020, apart from a period in Melbourne where there was a quite severe outbreak, and almost all of 2021 until Delta and Omicron. No cases, apart from ones that were kind of secure in hotel quarantine. And the single most important decision that was made by the Australian government in close collaboration with academia, because of years of working closely together and trust, was to close the international border quickly. Sorry, at the risk of sounding like Sir Humphrey, that was a courageous decision by ministers, but it was the right thing to do, and it was built on trust. I guess the second thing really is that what you're talking about is the use of, you could call it ambulance at the bottom of a cliff data, right? It's describing what has happened when it's too late. And I would say, I mean, this is not my forte, but I know that there are really innovative and game-changing things that we could be doing with artificial intelligence, with all of the data that we hold in Public Health Scotland, in academia, international databases, and bringing those together to be much more predictive rather than reactive in our response to outbreaks. And again, I will keep plugging this partnership, because that's really why we're here today, is I'm really hopeful that through this partnership we can bring together those data sets in Scotland, because goodness knows we've got such deep expertise here and we've got fabulous data. We should be bringing it together.
Great. Matt.
Yes, Mark. Just to pick up the point about genome sequencing, and again, I think that's a good example of a technology. In fact, COG, and I think Emma will correct me, was formed in the very, very early days, and the sequence for the first case in Scotland was within 24 hours, 48 hours was generated. So I think very early on we had a picture of it. That said, I would argue that that's a great example of where COG was driven by sort of key network of researchers with expertise that just got together very, very quickly under the sort of leadership of Sharon Peacock, with funders getting on board very, very quickly. Reflecting on that for how we prepare in the future, we've seen the value of it and we recognised the value we have capability in Scotland. But if Disease X was to still appear, that we would still need, there would be a lag in terms of firstly identifying it and working up all the protocols, and that's where the partnerships and the collaborations are so vital into providing the skill sets and the research sets that can then rapidly translate and use the capability that hopefully is the legacy of the COVID the experience. But it then requires systems to move in place, and we have to be better at that. And again, I think this is where the sort of the report that's been produced and the sort of network to actually identify how we can keep those links and utilize the capacity as you say, to provide data right from the beginning, not months down the line. But that's just the view from genomics.
Anything to add? Thank you. I think few things that we need to remember as part of our memory and learning. Human memory is very fickle. The faster we learn, the faster we forget. And today it looks like 5 years ago, pandemic didn't happen. I was part of the Royal Society of Edinburgh's post-19 COVID commission, post-COVID-19 commission. And in that, we did talk about, and the business resilience group and data and communication were key aspects of it. And if you go into the detail of it, when something like that happens, every board's or organizational board's priority is to bulk up a stock and keep things that are required. But as time passes and resources and cost cutting happens, those are the first things to go. Nobody keeps masks, nobody keeps ventilators hidden in the closet, nobody keeps hand gloves, and that thing will happen again. I think so. It's not that we can be prepared for everything. It's just the question of, do we have the sort of frameworks, and do we have the collaboration agreements between the organizations that need to come together? So if I take a live example of that, during the COVID-19, antibiotic use went up by 30%. World's various organizations have reported, BBC reported it, that the next silent pandemic waiting to happen is antimicrobial resistance. We are having so many superbugs coming. No new antibiotics have been discovered in the last 50 years or so. There are efforts being. And Anna mentioned microplate DX. The reason I'm saying is that we are developing a test where it takes 3 days to 5 days. We can do it in 1 hour. The challenge we find for most people in terms of funding or adoption is, will it be adopted? Is there a use we can do as we are doing? So the question is, how do governments prioritize it? UN has declared that there has to be death reduction by 10% by 2030. To do that, you need to apply the technology that's available to that, support it, or you need to change something in the regulatory pathway, and that would be diagnosis. But are we thinking that far ahead? So the question here comes is that what do we learn from it, and how do we prepare? And in your other point of it, in terms of early days, how many virologists who are experts who know the virus in the TB early days? I think most people were either modelers or thinking about, which actually statistical calculation is very different from how the biology works. That probably would have taken a lot of the fear away and maybe reality brought in there. So for future mix, having people who understand the subject matter in the mix and them talking to public on the TVs, as well as having the business people who are actually there to collaborate and support, will be the key messages that government and the organization need to think about.
That's a great message of Triple Helix. We have 2 questions there. I don't know who was first, but the one who gets the microphone, please.
Okay, hi. Steve Reicher, I'm a psychologist at the University of St Andrews. I want to pursue this issue of temporality, because it seems to me that actually temporality is critical to any collaboration. We often discover that cross-culturally. If I agree with you, we'll meet at 12, and I mean 12, and you mean sometime in the afternoon, we're going to fall out pretty quickly. And one of the things I discovered when I became involved in the advisory process is a question was asked to me, and I thought, oh, that's a good question. Well, I'll think about it. I'll write a research proposal. I'll get the research proposal. I'll start the research. I will do the analysis. I'll write it up. I'll get it published. I'll give you an answer in 5 years' time. And Daniel would say to me, well, we wanted it the day after tomorrow, but we'll be generous. You've got until next Tuesday. Right. So completely different notions of temporality in these different institutions, which get in the way. And so my first question is, how do you acknowledge those so that you know what the issues are? But secondly, how do we speed things up? In all that we do, and I start as an academic. So for instance, I wanted to do research. I do research on crowd and collective behavior after the 24 riots. So I went to the SRC and say, look, is there any way round the funding gap? And they said, well, that's a good point. No, there isn't. Okay, which is ridiculous because it means not just in terms of pandemics, but anything that happens, we can only research it long, long after. What about ethics? Our ethics procedures take a long, long time. So we need to do ethics well, but we need to do it quickly. Then what about things like reviewing processes, like publication gaps? How do we think about all our structures in such a way we can deal with those problems of temporality? Because if we're going to be prepared next time round, we've not only got to have the relationships, I completely agree with Deborah, we've got to start now in building those relationships, but we've got to think about structures so they're fast acting and they deal with these different notions of temporality which get in the way of collaboration.
Great question. They will all die to address it at the moment. I don't know who would like. Deborah wants to start. Go.
To be clear, I just want to furiously agree with you. But I mean, it's, you know, it's so true, having those structures in place, having those umbrella ethics ready to go, having the clear line of sight to regulatory approvals, having the, you know, the data sharing frameworks. I mean, they're critical, and I hate to say this, but I do think we have to look south of the border for some of their structures for how to respond. I was going to say rapidly, but quicker in this kind of setting. And you know, I think the NIHR-funded HPRUs are quite a good model for this, actually, because you know, they're set up around a number of themes, whether it's bloodborne viruses and sexual health, or foodborne disease, or you know, when I'm involved in public health genomics or emerging infections. And that is one of their raison d'etres, really, and there's one in behavioral science, is really to have those frameworks in place to enable that rapid exchange of data, exchange of funds, and exchange of knowledge. And you know, I think that there are definitely learnings from that, and I know, I mean, gee, it would be a fantastic model here.
Okay. Matt?
Yes, just to, again, agree wholeheartedly with that. I think there were a couple of things. I think the first point you made about an academic's perspective on research and the timeline and what Daniel was perhaps expecting, I think one of my observations about sort of working in collaboration very early on is just the different sort of cultures and expectations in academia versus public health, and when the two come together, it's an interesting mix. And I think this speaks to the point about, in terms of being future prepared, actually more opportunities for academics to sort of have roles and experience of working within government and policy and in public health agencies to make them aware of the requirements. I know, again, it was a very steep learning curve from myself to be dropped into that environment where things are expected. Next day, maybe there was a comment needed for First Minister's briefing required within 45 minutes. So it's a sort of a change from maybe taking 4 years to write that superb paper that you've been dying to write. So there's that element. But then all the temporality, I think Deborah's absolutely right. If we look at what were arguably the things we could have done better to facilitate the collaborations, it's around the sort of processes and speed of getting data, sharing data, the ethics, the PBPPs, and so on. Those are the things that we need to work and again improve. The experience with COVID rewrote the rules. It showed us what was possible, and then we changed our processes, but we've now reverted back to business as usual, and it has got harder to do a lot of these things. So I think it's important for us to revisit that. And I know in the plan there are specific goals in terms of processes for sharing data and making it easier, and that absolutely has to happen.
Great. Last word on this.
Thank you. I think that's a key point that you're talking about temporality, because it's a cultural issue. It's a business and academia work with two very different cultures. If you say business says we need to do it today, they will do it today because otherwise the business outcome depends on that. Whereas we are more flexible in academia with that timelines, with the grant deadlines, and unless it's a 12 o'clock midnight deadline. But that indicates to you while there is a difference. So, which is why for things like that, there has to be the interface of people who have worked in both, the knowledge and understanding of both cultures, because we are not going to change overnight the culture that exists, which is why they are successful the way they are. The research requires that creativity, that time to think about it, and business requires a task that needs doing, and then they move on to project. So having those interface of experts identified, having established framework which exists, and if need be, then brought about it. but also innovation hubs and multidisciplinary teams, which are there at the interface of these two things, which can actually expedite things that happens, because the viruses or the pathogens or organisms are much cleverer than us. That's the reason they have survived this long, and we don't know left field. We can predict it, but we absolutely won't know where it comes from. So we need to be prepared for the broader set of things that may expect. We will always need to change things. But then the question is, as I gave initially the example of engineering, ventilators, nobody was expecting McLaren or Dyson or the companies to make them. But they did come together and make together. And it's not that these things didn't happen. Vaccine, initially it was mentioned, it takes 20, 30 years. It was done in a year. So that is extraordinary celebration of that can happen. When ethics can be approved, or when the data, clinical trial data can be approved while it's being managed, so naturally it can happen in other time. Why should it take 3 years or 5 years at that time? So it's a question of bureaucracy, what is required versus what is actually minimally yes, and something that is built in there. So for those, we need to not forget what we learned. And with the AI and the data, now I think it's possible to expedite some of those timelines that you are mentioning.
Emma, do you want to add to this, or do you have a completely new question? It's related question. Related, because there is also a colleague at the back. Yeah? Okay, you go.
All right. It's for Dr. Malik, and I wonder what the opportunities are for people early in training, to train both in academia and entrepreneurship and innovation, and should we be starting much earlier with them? And what are the opportunities, and do we need to expand them? That's
absolutely. I think, as I said, entrepreneurship is not just about the company formation. It's about seeing the opportunity and learning to take the risks and seeing to say where else it can be applied. So starting even at undergraduate level to say what's the difference, what's the commercial potential of a knowledge that you know, and which is why it all boils down to intellectual property. So when people start to learn that difference, and because in academia we are very good at disclosing everything to everybody, and sometimes that becomes a problem, which is why we need to learn and backtrack and train academics to say don't disclose this, because then it becomes the thing that you cannot patent it, you cannot. And science based on deep tech, certainly you can do startups which are work not deep tech based, but in this case certainly there is a potential for that. Oh, absolutely. So it means doctoral training in there. I would say, as I said, when I said as early, now we are talking about introducing in schools. But that difference of what is an opportunity, what are the basic things that are required to take it into the market, understanding, so you start. But PhD, certainly doctoral training programs. That is not too early. That is what you are doing, your research, and you need to think about what is the potential. And it's not for everybody. But then like that, academic research is also not for everybody. And the other point which I will bring into it, and which is another favorite passion, is the lack of diversity in women founders and leaders in business sector, because not many postdocs, if you will say, whereas in science and in research and in health sector specifically, there are so many more girls and women. Why do we not see them finding companies? Why don't we not see them as investors who can allocate? There is plenty of research shown. It is because we don't encourage them and they are not. So it starts at that level and going for that. So yeah, absolutely.
Okay. We're sort of running out of time, but I need to add a little advertisement to this. On the 28th of this month, 28th of March, Academy of Medical Sciences and Chief Scientist Office together will launch a hub, AMS Hub for Scotland, which will be addressing particularly these issues. This is for young people to mix interdisciplinary, both entrepreneurs and PhD students and postdocs, but from many disciplines, to come with projects. They could be in pandemic preparedness, they could be in anything really. So if there are still a few places left, if anyone would like to come, Stephen Kelly at NRS is the man who can open the door to this. And we have, Andrew, a few places. I think this is really an interesting joint enterprise. The money comes from the Wellcome Trust, I understand, and there is money for projects. So young people could come, form teams, and then do things, which is fantastic. So delivery, delivery, delivery. Thank you. There was a question there for a long time. We need to...
Thank you. I'm Rowena Neville, a GP, and I was really interested in the global experience that you have on the panel. I was working in Jordan in a refugee camp when the pandemic broke out, and I was just wondering, do we have now consensus on what was the best kind of approach? Because there were, like you said, so many different approaches. Jordan, again, locked down instantly and kept this zero COVID policy, and we were looking in horror at what the UK was doing. Do we have people analysed not just the numbers of infections that were allowed to develop, the numbers of deaths, but then the impacts that these very strict lockdowns and closing the borders had? Because that feels like everyone was unclear at the time. Have we analysed and learnt from what we should all do from as early as possible in future?
Thank you. That's a great question. I think Deborah starts on that one.
Oh, I was just looking at Jim sitting here, thinking about whether to pass it to Jim. Yeah, I think the data are still emerging. I think I mean, you mentioned the impact of lockdowns, and I don't think we've seen the full, I guess, the full impact of that. School closures, etc. Long COVID, we heard about. I would just say that from, if you look at public health infrastructures, countries that, and I hate to say did well because it's not the right terminology that we should be using, but countries that I guess were resilient through the pandemic, I guess, were countries where there was a really strong public health infrastructure in place already. And people often say South Korea is a good example of that, Taiwan, for example. And again, if you, you know, in my role in Australia, we were involved in discussing with some of these countries, and, you know, it comes back to this collaboration and trust. You know, they had this multi-sectoral approach. Industry was right at the heart of what they did as well, industry and technology. So, I mean, I'm sure that there are others, and, you know, we've got the CMO here as well. Sorry to throw you under a bus, CMO, but, you know, we've got others in the room who may wish to comment on that, but, yeah.
So yeah. Absolutely, absolutely. No, seriously, Gregor, Jim, all comments welcome, but microphone, yeah.
So Anna, I'm going to give some closing remarks, or maybe save my comments to those closing remarks.
Okay, that's fine. Jim? Jim?
Passing the mic to Jim, there's like an opportunity.
Thank you. I agree with everything that Deborah said, that the jury's yet out about what package of things combined offered the best opportunity and worked in practice. I imagine that it depends on the endpoint that you want to look, and that much of the discussion in the background has been over what time frame. We've got the immediate 5 years that we've had. What's going to be the lasting legacy issues that we see that are harder to measure, whether that's because of the lockdowns that were implemented? What does that mean for educational attainment? What will it mean for some of the long COVID things that we've discussed? So it is quite a complicated area.
Thank you very much. Could I thank the panelists? I think they've done a great job today. I think we heard things that are important at this stage 5 years on. I would like to thank for all the questions, which were very important and touching on things that we need to know and think about for the future. So thank you all, and could we thank the panelists in the usual way? And we are before time.
Wasn't that terrific? It was great. So we're on the final furlong, and it gives me a great pleasure now to invite my good friend Gregor Smith, who's the Chief Medical Officer for Scotland, to make a few closing remarks. So please welcome Gregor.
Hasn't this been a fabulous afternoon? And the quality of both the presentations that we've seen, but also the panel discussion that we've heard, has been, I think, fitting for the importance of this topic. It's a real pleasure to stand before you this evening, just to speak a little bit and offer some closing remarks before we finish tonight. Right at the beginning, Andrew asked us to do 3 things with today. Andrew asked us to celebrate the report, and I think that's really important. This is a moment in time that we need to build on. The publication of the report is not the destination, but actually it's making everything that's contained within the report, those 5 recommendations, happen in the real world. That's, for me, the journey that we now need to embark upon. And we've heard the word collaboration used many, many times today. I think that's absolutely critical. It was a feature, so strong a feature of our pandemic response, but actually we need to make sure that in this next stage, that collaboration holds true. And I'm excited, I have to say, by the sense of the Scottish Pandemic Sciences Partnership that we've discussed on a number of occasions today. I think that's a critical component of how we make sure that we're best prepared for the future. Andrew also asked us to kind of celebrate the great science, and my word, have we heard some presentations today that give us optimism for actually the science that is going on in Scotland. It's no surprise that actually the areas that we did fare better with over the course of that pandemic response was where science in the UK and in Scotland in particular was already strong. But we need to build on that and to make sure that we broaden where that scientific expertise lies as well for the future, if we're to be truly prepared for the next pandemic when it comes. Like many people you've heard speak today, I think it's inevitable that we're going to see a pandemic. I think it's much more likely that we'll see another pandemic sooner than the gap that we saw between the previous experiences. And in doing that and acknowledging that, we just have to make sure that we prepare appropriately. And Scotland can contribute in this stage. We've already heard some of the wonderful examples of the work that has gone on here, that has developed, and that we need to kind of further build on. And the last and third thing that Andrew asked us to do is to look forward, and we've heard us begin to do that. In looking forward, though, we need to make sure that we're also looking back and that we're learning from our experience. The report from the Standing Committee, I think, is a moment in time. We add that excellent report to other sources of learning from that time. There's any number from the Royal Society here, from other institutions. The 4 UK CMOs have contributed into this space with a technical report, which I think is equally important that we use to learn from. And of course, the COVID inquiry continues even now with hearings in London as we begin to develop the broader learning. And all that taken together must be kind of encapsulated and built forward. And science has to be at the centre of that. But science alone is not going to be enough. Science has to be there with the policy to support it, with the infrastructure. I've heard so many times today about the infrastructure, but with investment in the infrastructure to make sure that we are able to respond in the way, whether that be through the public health community or whether, just to get my own little dig in here, is a national public health reference laboratory, capitalising on the system of laboratories that we already have, but making sure that we take advantage of all those assets across the country facing in the same direction that are going to be scalable when the balloon goes up the next time. That, for me, is something which would be unforgivable if we don't take advantage of for the future. So making sure that we've got that infrastructure, that we continue to make that investment in that area is absolutely critical. But let's not forget the human impact. And we heard the Cabinet Secretary at the very outset of his opening remarks talk about the very real impact that it has had on our population, the people we've lost, the people who continue to experience incredibly debilitating symptoms as a consequence of their exposure to COVID disease, and also the impact that it's had on those of us who were involved in responding to it, whether those were clinicians in wards or GP surgeries across the country, or whether they were involved in some other public health part of the response. The pandemic has left its mark on all of us. Too often today, I've heard us refer to the fact that larger parts of society have perhaps forgotten about some of the impacts. It's right that we remember those impacts. It's right that we begin to build a system in place so that we can respond the next time it's necessary. There's so many people I would want to thank today for putting on this event itself. I want to make sure that the work that Marianne has done is acknowledged, and particularly in this space, because I think this is really important. I want to thank all of you for your contributions today, particularly our speakers and our panellists. I think this has been a really important aspect of the day, to hear directly from the people at the centre of this science. I want to reserve a special thanks to Andrew for the way that he not only led, first of all, the COVID-19 advisory group within Scottish Government, but then the Standing Committee on Pandemic Preparedness afterwards. Andrew's a remarkable man. He'll hate me for saying this. There are very few people, I think, could have brought together with such credibility such a wide group of scientists, researchers, clinicians, policymakers, and managed to give us a product that was incredibly helpful in terms of shaping our pandemic response. I remember having a conversation with my predecessor, Catherine, when we were trying to identify a chair, and I think we both landed in the same name almost immediately at the same time. And Andrew, my eternal thanks for the support that you gave me during that pandemic response as well. So collaboration, ensuring we've got the infrastructure, whether that be the pipeline of early career researchers, people who are attracted into research for the future, all of these are absolutely important. We need the stability to be able to do that. But we always need to remember this, the human factors in all of this as well. So one last word in all of this that I haven't heard today. And I want to introduce this into people's thoughts. We've spoken about the importance of science in responding to the next pandemic, but really there's so much more that we need to start doing now if, as a nation, we're ready to face the challenges. And right at the centre of that is to identify and reduce the health inequalities that exist across our country just now. We've spoken about resilience a lot, but ensuring that the health of our nation is more equal and is of a higher standard than it was when we entered the 2020 pandemic has to be an absolutely critical point of any preparation for the next pandemic. I don't think that it's any coincidence that some of those countries who fared better, for want of a better term, actually had a pre-existing health within their nation which was of a higher standard than we've been able to achieve in Scotland. So supporting our population to achieve that higher health is something which we can begin to do now and must do now, and close that health inequalities gap that exists across our society. So thank you for your attendance today, for your interest in the work that's going on here, and for those of you actively involved in research and the development of our future pandemic response, my eternal gratitude for that. Thank you, Andrew.
Great. Thanks, Gregor, for those thoughtful words. Do you want to take questions? Anyone got a question for CMO? You've blown them away. So there we go. So, okay. So, yeah, a few reflections. Yeah, just to echo Gregor's thanks. First of all, because the Royal Society of Edinburgh, hasn't it been a great, it's a good sized meeting, isn't it? I always think meetings 60 to 80 people, it's great. Apologies to the folk online who haven't been able to ask those tricky questions. So, but we'll get that right next time. But thank you for bearing with us, folks online. But Leslie Ann Lamont, can we give her a round of applause? Because she's been great. So, and actually, it's really important to shout out Marianne again, and Caroline, Daniel Kleinberg, and also Jerry McLaughlin, who retired from Scottish Government after, he says, I've been working here 400 years. But Jerry, there was a team that actually, not only has been the steward of the C19 group and the Pandemic Preparedness group, but has always been on call to really provide a very human face to government, and it's been fantastic. So thank you. It's been a real privilege to work with you. And today, can we thank the Cabinet Secretary? He stayed for the science. Wasn't that great? Most ministers, they say, thank you, good night, and they're out. So all credit, he stayed for the science. We should acknowledge that. But also Paul, Whale, Clara, Mark, Alice, Matthew, Poonam, Deborah, Gregor, and Anna, who I thought it's been a great afternoon of science. So maybe a round of applause for everyone. So that's great. And then, and lastly, I think Gregor summed it up beautifully. We've got to look ahead. We've got great science, and the people today that covered. We went from mRNA to temporality to pregnancy to long COVID. It was a great spectrum. But we talk about the strength of the science, but it's really the strength of the people who's doing the science. And I think we're in good hands in Scotland from folk that we've got. But it is that collaborative partnership that we mustn't take for granted. So how do we nurture that? And looking ahead, we're still learning from the pandemic, but also, the world's a pretty tricky place in terms of NHS pressures, as Gregor says, inequalities. The economic outlook is challenging in the UK, and there's global sort of geopolitical instability. And the voice of science and a trusted voice is going to become increasingly important. So to finish with two quotes. One is, don't mistake activity for achievement. So that's, and I would challenge us all to subscribe to coming back in a year's time or in 18 months' time when the partnership is up and running, and maybe hosting its first symposium where we are celebrating progress, these relationships that Deborah talked about, so that they're business as usual rather than exceptional. So I think we should challenge the partnership to say we're going to support you, but let's come back and have a similar type of meeting. But as we look forward, I always think hope's important. So because once you choose hope, anything is possible. And I think we should be optimistic for Scotland because there's great talent. And we have that social contract with the citizens that we shouldn't take for granted, but actually can propel advancement in this area. So I think we've had a great afternoon. Can I thank everyone? Thanks, Anna, for plugging the Academy of Medical Sciences. That was very great. You were very good at promising our money to other people. So we'll see you all in Glasgow. It's next Friday. Yeah, it's next Friday. So we're coming back. So, and if anyone wants, take risks and do good things. And we'll see you in a year or 18 months' time when we'll celebrate success. So, thank you all, and see you all soon. Thank you.